Okay, good afternoon and welcome to the second day of our TPCD-1 webinar. To all the panelists, thank you for sharing your time and expertise for today's two sessions. The University's Hassanuddin for hosting, Jinyalg and Safe Seaweed Coalition for the financial support, and Dr. Alan, who led in organizing this webinar. It's really amazing to see and meet all the seaweed experts from the temperate to the tropics gathered since yesterday to shed more lights about their presentations and learn lessons from each other. Thank you for all the panelists and the facilitators. Today's sessions are equally interesting and engaging. So I encourage then all participants to join the QA and chat boxes for questions or comments, for interactions and active participation. It is with high hopes that this seminar will spark regional and international collaborations. Encourage further our colleagues to stay connected with one another even after this webinar. Again, welcome everybody to the second day of our TPCD-1 webinar. We will start today's session with a plenary talk from a friend and a colleague in the world of euchymatoids in the person of Dr. Ian Neese. Dr. Ian Neese is a Canadian phyconomist who has followed the path of his father, Arthur Charles Neese. He commenced a phyconomy vocation with commercial seaweeds in 1965 in his home province of Nova Scotia. He has worked all over the world, but since 1977, his work has been based variously in the Southeast Asian region, namely the Philippines, Malaysia, and Indonesia, where he lives and at present works with C6 Energy based in Bali as the director. The core of his phyconomy practice has been adaptive development of euchymatoid seaweed value chains from within the private sector. Let's all welcome Dr. Ian Neese to talk about BBNet, a window to real problems of euchymatoid farmers. Ian, your video.
Okay, let's see the video.
Your turn.
Somebody there has to push a button, I guess. Hello? Here we are.
Is there a sound?
Yeah, there's no sound.
Hi, we're showing the video. Could we check the sound? Okay, Ian, you might have to do a voiceover.
Hello, my name is Ian Neese, and I'm an adaptive phyconomist, a seaweed farmer. Bibit is the Indonesian word for vegetative seaweed propagules, otherwise known as cuttings. These are often erroneously referred to as seedlings. So what is BBNet all about? BBNet will be a seaweed industry stakeholder collaboration that can alleviate farmer risk and enhance farmer rewards in Indonesia and other tropical countries. BBNet will support networking and capacity building of existing organizations. It will not reinvent the wheel. One for all and all for one. BBNet facilities will produce both goods and services. BBNet facilities will comprise sterile cultivar banks that feed biomass through nursery systems to commercial farms with farmer planting schedules in mind. BBNet goods will comprise healthy and certified biomass that comes as close as possible to meeting farmer and processor needs. And BBNet services will ensure proper management and specification of hatchery and nursery stocks, traced through entire value chains. What are BBNet attributes? BBNet will blend precision and adaptive phyconomy. BBNet will catalyze consolidation of the cultivar core that must be a foundation of responsible, profitable tropical phyconomy. BBNet must integrate into farm ecoscapes and local ecosystems. The BBNet system must be sustainable and anti-fragile. Cultivar identities should be traceable and certifiable from source to end user. Cultivar biomass must be productive and profitable as commercial seaweed crops. And cultivar types must be practical and robust as biomass foundations for seaweed farms. Cultivar should enhance food security and biosecurity of seaweed farms. Farmer needs and biomass source analyses are ongoing as a first step in building BBNet. So where in Indonesia is seaweed farmed, and what is it worth? Seaweeds have been farmed in most of Indonesia, as you can see in this map. Industry estimates of production for the three major farm genera is on the order of about 12 to 15,000 metric tons per month of Kappa-ficus, 10 to 12,000 tons a month of Gracilaria, and 2 to 3,000 tons a month of Eucuma. So that would be a total of about 25 to 30,000 metric tons per month. About 50% of the Kappa-ficus is farmed in Kalimantan Utara. About 25% of Kappa-ficus and 80 to 90% of Eucuma is farmed in Sulawesi. About 25% of Kappa-ficus and around 10 to 20% of Eucuma is farmed in areas throughout Indonesia. Most Gracilaria is farmed in South Sulawesi and Central Java. The area enclosed by the dotted blue line here can be tremendously productive in East Indonesia, but logistics costs impair links to value chains from that area. Confirmed data are unavailable, but informed estimates place current production volumes in the range of about 300 to 360,000 dry tons per annum for all of Indonesia. Now this is by informal industry consensus. By official government estimates, current production volumes of all types of seaweed from Indonesia are on the order of over 10 million wet tons per annum, and that would be over 1 million dry tons. That level of production puts about $500 million US, more than 7 trillion rupiah, into the accounts of seaweed farmers around the seashores of Indonesia. So what farmer needs would be met by BBNet? Well, to begin with, upwards of 50 to 60,000 Indonesian farm households need what BBNet proposes to offer. One of the most important is cost-effective recovery from seasonal crop failures, which are often associated with the ice malady. Some BBNet benefits? Well, there's best biomass for farm initiation, optimum crop mix for best prices, and rapid recovery from crop failures, as I just pointed out. In addition, farmers should get more crop cycles per year, better productivity per cycle, and millions of rupiah higher earnings annually. The bottom line is billions of rupiah in annual income for farm families. What cultivars would be produced by BBNet? Well, there's an expanding selection of globally available red, green, and brown seaweed cultivars. There are many cultivars that exist, are being commercially farmed, but are not readily available to farmers around the world. In all cases, with BBNet, cultivars would be identified and traced using molecular taxonomy techniques. What is the timeline and budget for BBNet? Well, BBNet is currently seeking funds for a 2-year, $250,000 development plan that would build on existing facilities and nursery locations. BBNet development is tied to Tropical Phyconomy Coalition development, as manifested by the workshop for which I made this presentation. You can follow some of what's going on by looking at the website at phyconomy.org. Please help us to build the BBNet initiative. Thank you for watching this presentation.
I would like now to turn over the microphone to the moderator and facilitator for the remaining of this webinar session.
Thank you very much. And along with my colleague, Dr. Inaya Yasir, I am Sudirman Nasir from Hasanuddin University, will facilitate our discussions this afternoon. And before we discuss further with other panelists, we are going to focus on dialogue with Dr. Nash in relations to the just presented presentations on BBNet, a window to real problems of accumulate farmers. And with us at the moment, there are several questions that we are going to ask. Actually, the questions were asked by the participants, and we hope that Dr. Nash will provide further elaborations. I will choose one of the questions. Dr. Nash, one of the question is, how will farmers from various regions engage with BBNet? And how, for example, the farmers stay in touch with one another, and how can they get daily prices or values for the sale of their raw materials? Could you please elaborate and respond to the questions?
Yes. Can you hear me?
Yes.
Yeah.
It is clear.
Okay. Well, first, I would like to say that the first step in making BBNet happen is this webinar that we're involved in today. Now, we were discussing how to make BBNet happen, and it was obvious that we needed to come from two ends of the spectrum, so to speak, from the precision phyconomy end with the academics and scientists and so forth, such as the ones we have involved in this webinar. At the other end of the scale, we need to engage farmers. And that is the next step that we will follow up on after this. Now, we've been engaging with farmers through Jasuda.net. I should have put the logo of Jasuda behind me today too. But our first method of contact with farmers throughout Indonesia is through the Jasuda network, through which we get data on prices and so forth from all over Indonesia all the time. And through Jasuda and focusing on various areas where we are very active and Jasuda is very active, we're undertaking basically a needs analysis based on discussions with farmers. Now, I spend as much time as possible in the field myself. And after all of this webinar stuff is over, I plan to get on the water and stay there as much as possible, going to farmer communities and assessing their needs. And basically, that's how we bring the farmers into it. In future webinars, I hope that there will be farmers involved, farmer group directly interfacing with the academia, bureaucracy levels that we're dealing with now.
Thank you, Dr. Leis, for these responses. And there are some follow-up questions from the audience. And one of them is the questions that asking about what are the primary factors affecting how much a farmer can get paid for the product. Is it a case that they can hold materials, or do they need to sell regularly to maintain a cash flow?
Well, what can I say? The seaweed market is an open, free market. Prices are not fixed. They are determined by the marketplace. The law of supply and demand is the overarching law that governs seaweed prices. I know farmers always complain about the price. Any farmer anywhere in the world farming anything, the first thing they will do when you interview them is they will complain about the price.
Yes.
So that's a given. Farmers will never be satisfied by the prices. But I think it's essential. You know, Dr. Jompa was talking yesterday about the pentahelix and was asked what's the main factor, most important factor, and he said industry. Well, I would go further than that, and I would say that value chain players must make a profit. The value at all level of value chains, every player has to make a profit, or the value chain breaks down, and then there is no seaweed business.
Yeah, there's
So
also. Yeah, please continue.
So farmers have to make a profit. They can do that partly by getting a higher price and partly by being more productive so that their cost is lower. And that's what makes their margin.
Yeah. Yeah, there's also questions appear in questions and answer room about how this kind of initiative can be expanded to farmers in other countries, such as countries in South Asia, including India. How do you respond to that?
Well, I guess I got involved in spreading Southeast Asian technology to India by working in India from 2000 and 2004 in a project that was going on there. So it's really people like us who are participating in this webinar who are responsible for spreading around farm technology. Because it's not actually us scientists who have developed what I call adaptive phyconomy methods. It's really farmers who have adapted the methods. And we have been lucky enough to be able to travel around and meet farmers all over the place and record what we see different farmers doing. But honestly, I think it's true to say that every commercial cultivar of eucumatoid seaweeds known today was found by farmers. And that the practical phyconomy by which they grow their crop was developed by farmers. And the main purpose that people like me have had is to connect among the farmers by traveling around the world and meeting farmers all over the place, and also by writing, manuals and conducting courses, which I have done with Ann Hurtado and very many other people over the years.
Okay, thank you, Dr. Nesh. And now from India, we move to questions from Indonesia from one of our participants, Pak Aslan, who mentions that in Indonesia's context, businessmen, academia, and also government agencies, particularly those from Indonesian Ministry of Marine Affairs and Fisheries, frequently discuss about production in relations to Indonesia's seaweed production data. And from those discussions, it was found that there is an urgent need for revisions of the production data because it tends to be higher than the real data in the field. And this is an important system that need to be reviewed to maintain sustainability of Indonesian seaweed productions. how do you elaborate or respond to this phenomena that perhaps not just uniquely in Indonesia, but also apply to other countries in Southeast Asia?
Oh, okay. Well, I'll talk about the world. Of course, this is a tricky thing to discuss because I am a foreigner who lives and works in Indonesia. So discussing what the Indonesian government is doing is, well, they've been very supportive and helpful, but I must be careful what I say. But as I pointed out in the video that you just saw, there are government figures and there are industry figures, and the two are very wide apart. Roughly speaking, the government estimates of production are 3 or 4 times higher than the estimates I get from industry. But the fact of the matter is that nobody is out there actually measuring seaweed production. So we're dealing with estimates. And that is true worldwide. If you look at the FAO numbers, those are numbers that are given to FAO by the different the different countries that are involved in FAO. And as far as I know, almost all of them are estimates. Now, there are exceptions. The one exception I'm familiar with is in Zanzibar. There's only one port. Everything gets exported through that port. And therefore, they know exactly what is being exported. And they record those exact numbers. That's the only place where I've worked where I really, really knew what the production was.
Dr. Nash, and there's one more questions from the audience from participants, Peter van der Hayden, asking about the Seaweed Farmers Associations in Indonesia. And as we know, there are many in several regions in Indonesia. And can you mention some of those who are very actively at the moment? And how do you think the role of these associations can be further enhanced?
Well, they do a great job in pulling things together. For example, Arlie, which is run by my longtime friend, Safari Aziz. He's a real pioneer in developing seaweed farming in Sulawesi and actually all over Indonesia. And so Arlie is great at handling industry issues at large and so forth. And then there's Apdirli and various and sundry other official organizations. But the thing is that, you know, almost every farmer belongs to some kind of kelompok or group. And then those are in turn are kind of consolidated at the community level and then at the kabupaten level and so forth. And in pulling together farmer interests on that, I must give full marks to KKP and DKP, the fisheries ministries throughout Indonesia, who have a lot of people very active, integrating with farmers and performing a very constructive role in helping this whole industry to develop. And it's been that way ever since we started back in the 80s. We've always had, from my first meeting with Dr. Habibi in 1988, he said that they would provide support at the government level, and they have delivered on that promise.
Thank you again for the responses. And now I think we are going to move to other panelists in these third sessions. They're focusing on lesson learned and also roadmap for further development in tropical Phyconomy. But before we move to that, I would like to invite my colleague, Dr. Inaya Yasir, to introduce all panelists in this session. Dr. Inaya, time is yours. Once again, thank you very much, Dr. Nash.
Hey, you're welcome.
Thank you, Dr. Sudirman. Hi, everybody. I will introduce our panelists in session 3 today. Yes, the first one, I think Dr. Anicia already introduced Ian Nash, and nicely. And then the second panelist will be Sylvain Faugernon, a professor and the president of the Chilean Phycological Society. His research is combined ecological experiments and population genetic. Our next panelist will be Juliet Brody, a merit researcher in phycology from Natural History Museum in London. She is an international expert in seaweed with over 35 years experience. She focused on seaweed in a time of rapid environmental change, including a citizen science seaweed program. Next panelist. Next panelist is Jonas Collin. He is a researcher at the Station Biologique de Roscoff, France, with a research interest focused on physiology, ecophysiology, and genomics of intertidal seaweeds. Our next panelist will be Simon Davis, founder and managing director of Seedling, an environmental and social impact seaweed biotechnology company. He is a firm believer that responsible business is the key driver to creating lasting sustainability, and is a passionate advocate for the pivotal role that seaweed will play in generating novel products for our future, whilst combating carbon and restoring abundance to our oceans. Our next panelist will be Paik Im Lim, a professor and deputy director of Institute of Ocean and Earth Sciences, University of Malaya, a founder member and the current secretary of the Consortium of Southeast Asian Seaweed Taxonomy. She is involved in multidisciplinary approach, is adopted to address key issues. And she is involved in the Global Seaweed Star program. Next panelist will be Michael Y. Roleda, a professor at the Marine Science Institute, University of Philippines, Diliman, Quezon City. Experience with tropical, cold temperate and polar seaweed. Currently applied research collaborations, including using seaweed for food, feed, biofuel, and biopackaging applications. The next panelist will be Zanny Langford, Research Fellow in Rural Development University of Queensland, a social scientist and economist with an interest in land, food, and rural livelihood in Indonesia, Northern Australia, and the Pacific. Next will be Anicia Hurtado, who's already given a welcome speech early in these sessions. She's a chair of the Integrated Services for the Development of Aquaculture and Fisheries. Currently, the developing countries partner for Philippines for the Global Seaweed Star project. I think all have, and I hand it back to Dr. Sudirman to start the Q&A session. Please, Sudirman.
Thank you very much, Dr. Inaya, for introducing all panelists in our sessions. Before we facilitate further discussions, I would like to once again express our gratitude to all speakers who come from different regions, from Indonesia to the Philippines and Malaysia, and then across Arafuran Sea to Australia, and also to Latin America, in Chile, and then to Europe, in UK, and also France. Especially those who are attending our current webinar at Ortstein, we particularly express our gratitude. The different time zone will, of course, barriers in the current approach using digital technologies for our discussions. But COVID-19 pandemic that we all face at the moment also facilitate not just crisis, but also opportunities to push us to use technologies in facilitating academic and professional discussions such as our current event. So we are going to allocate time to discuss with all of our panels, and their presentations, as we all know, have been pre-recorded, and this opportunity will be asked for all of panelists and audience to be engaged in better understanding the problems. So I would like to start with our panelist, Dr. Sylvain in Chile. Hi, Dr. Sylvain.
Hi.
Yeah. So, yeah, of course, there are a huge time difference between Indonesia and Chile, and we really appreciate your availability and also your enthusiasms to attend our sessions. And there are already several questions asked by our participants. One of them from Alan Critchley, asking that if you had to select one restorative actions, what would be your top piece of advice as where to start for the greatest impact?
So thank you, Alan, for this question. It's a complicated question to answer because there are many problems to solve, and there is a lot of things we still don't fully understand about seaweed production. In the case of the Gracilaria production in Chile, we have learned at least that there is a major issue with the amount of genetic diversity in Chile. There was, before cultivation, there was a strong effect of over-harvesting that removed most of the genetic diversity. And now, if we combine this with the current cultivation practice that is based on clonal propagation, then you have very homogeneous farms in terms of genetic diversity. And this is really one of the main factors that is promoting the decay in the quality of the production, the invasiveness of pathogens and epiphytes. And so probably we need to learn to develop some better strategies to manage this genetic diversity through breeding programs or other cultivation practices that could use better the natural diversity available, for example. So that's probably one of the actions I would try to promote.
Yeah, thank you, Dr. Sylvain. And another follow-up questions from our audience is asking why diploid populations is more preferred over haploid for farming, and does it have any genetic advantage?
So in Gracilaria farming in Chile, which is done by clonal propagation, so the people harvest the biomass and use part of the biomass just to restock the farm. we have found that most of the farms is completely dominated by diploids. And actually, we don't really know why. The farmers cannot differentiate diploids from haploids, the sporophytes from the gametophytes. So they are not actively selecting for that. There are no known differences in terms of quality of the agar. And when we try to measure some physiological aspects, for example, we know in the laboratory that the sporophytes grow a little bit faster than the gametophytes. But when we look at natural populations, for example, females grow at least twice as fast as diploids. So we don't really explain why diploids are so much successful in the farms. It could be from some kind of advantage like the hybrid vigor, the heterosis that we see quite often in plants, in plant cultivation. So that means that you have some genetic properties of the individuals that produce a kind of vigor. But so far, our experiments have failed to demonstrate this existence. So it is something that we don't understand. It is one of the things that we don't understand, actually.
Yeah. Very insightful elaborations. And one more questions for Dr. Sylvain before we move to the next panelist. One of our participants asking about what farmers do during an epiphyte attack at the moment, and how do they find sources of epiphyte-free biomass to restart cultivations?
That's a very good question. So, what do they do? Usually they stop producing when the price that they get for the biomass is getting too low because the quality of the biomass is too bad. For example, when there is too much epiphyte in the gracilaria production, then a large part of the biomass is not gracilaria, it's something else. Usually many different species are epiphyting the gracilaria. So the quality is decaying, and then the producers have a lower income from selling their production. So that stimulates them to remove the farm and eventually start from scratch, from new plants that they buy. But where do they buy this new material? Usually from the neighboring farms. There are not, in Chile, there is no company or organization producing clean seaweeds, clean and healthy seaweeds that was mentioning Ian in his presentation. There is a strong need of clean, healthy, secure production of juvenile plants to be used for setting new farms. This doesn't exist. So they buy from the neighbors. They have for 1, 2, or 3 years of really relatively good production, and quite often after that, the quality starts decaying and they start over. So it's not really a highly productive way of managing the production, I think.
Thank you once again, Dr. Sylvain from Chile, addressing 3 questions. Perhaps you are going back to you for more questions after other panelists. We are now going to move from Chile to London and invite our colleague, Professor Judith Brody from National History Museum, who presented in the pre-recorded presentations about the microbiome and holobiont lesson learned from one carrageenophyte chondrus. And we already had some questions for you. And one of them is from a participant asking, how easy are the technologies required by microbiome studies transferred to the field? And can countries affected by imbalances in microbiome undertake their essays? Professor Vori, time is yours.
Yeah, thank you very much. Yeah, that's a question that's been exercising my mind for a while, and I think that is at the moment that's tricky. I think what we need to do is to understand very well the whole process of what goes on over the period of time that perhaps plants are grown, either in culture or in the wild, and then try to really figure out what's happening with the plants and the microbiome, what is affecting the plants through the microbiome and vice versa, and all of that. But ultimately, you know, these technologies are getting cheaper, they're getting easier. We're developing more protocols, and I can't think of a timeline, but I would like to think that down the line, you know, we can develop some tools and some technology that's cheap to use, that's simple to use, that takes this sort of whole understanding of the holobiome, but perhaps simplifies it down to some things that are practical in the field and that can be used. But at the moment, that, in my head anyway, others may know better, this is all a bit hypothetical, and we've got a lot to learn. But I do think we can learn a lot. And, you know, for those of you that saw the talk, you'll see, you know, how important it is to really know everything that's going on in the life history, the life cycle of all these plants, whether they be cultured or whether they be in the field.
Yeah, thank you. And as you all know, microbiome topic are now increasingly become the focus of research in many different fields, and including in seaweed. And another question, and this is asked by Dr. Nash, about your thoughts on the phenomenon of the ripening, or the so-called the Nash effect, how it applies to ecomatoids and other tropical red seaweeds, and how it may impact microbiome, epiphyte, and endophyte phenomenon.
Sorry, I missed that bit, the bit about the effect. Could you just say that again?
Yeah, the Nash effect, during which thallus color change and presumed to be related to nitrogen availability.
Oh, okay. Yeah. I don't know I don't know the answer to that question. We haven't looked specifically at nitrogen availability, but I think that is going to be something which it will be another factor affecting the microbiome. I mean, one of the things that we are doing a lot of work on, one of my postdocs who generated the microbiome data, Georgia Ward, she's been looking very closely with our global seaweed partners in Malaysia and the Philippines and Tanzania at how the microbiome changes in relation to the ice and trying to sort of figure out exactly what's going on there, for example. So I don't know the answers to those questions, but I think that they will in time all become apparent, and these are areas that we need to focus on more specifically. At the moment, what we've been trying to do is to sort of get an overview and to work out the methodology. And I think now that we've got that a bit more sorted, we can start to sort of delve more into these questions and then again, try to find some solutions.
Yeah, thank you, Professor Brody. There are, of course, several unknown territories in this field that require further studies.
Yeah.
And one more questions for you before we move to the next panelist, and perhaps you can go back later if the time allows. One of the participants asked about prebiotics. And as we know, that prebiotics are often used to influence microbial communities. Do you think this is possible with seaweeds? And similarly, prebiotics are often used too for human and animals' digestive tract. What chance do you think is there of introduced bacteria which may change populations or bacteria which benefit the crops of seaweeds?
Well, I mean, there are people who know more about this than me, but I think already people are using seaweed extracts to prepare the juveniles before they're put out into the field and so on, to try and improve their chances. So already people are doing that. I think some of the interesting areas here that I've thought about, perhaps a little bit separate to the prebiotics, is thinking about one of the species I mentioned very briefly in the talk. So this is the group of the Osmundias and the Laurentias. You know, they have these secondary terpenes, and I think those are very interesting, perhaps in relation to thinking about those in terms of maybe control within farms. So I think there's sort of some very interesting angles, like the prebiotics, and I did wonder as I was doing this work with chondrus, whether that has the potential to produce something that could be used as a prebiotic. I don't know, years and years ago, I did some experiments in the lab, and it does have an antimicrobial effect, and that probably is linked to the microbiome. But I think these are all areas where we need to invest in a lot of work. And there's actually one more point I would just like to make with all of this, is that you know, one of the things we have to do to be able to do any of this, we have to think very, very carefully about conservation, both of wild stocks, in situ and ex situ. And this picks up again with the things that Ian was talking about. You know, we have to secure what's in the wild, and a lot of this is disappearing, and there needs to be a concerted global effort for seaweed conservation for the future, I think, of the industry in many ways. And that's both in the field and I think outside the field. So I think that's the point I want to make. There's many, many angles to this, but people are welcome to put stuff in the chat. And I've got lots of ideas, but I don't know all the answers at this stage. I need people to work on that.
Thank you very much, Professor Brody, for enlightening us with some of the latest development in relation to microbiome, and also once again our appreciation for your time. And you mentioned previously about the soccer or the football team, and I just saw my neighbors here around my house who are the supporters of England, and some of them also Italy. So we are waiting for the very exciting game in the next few days. Thank you once again, and we are going to move to Dr. Jonas Cullen.
Yes, I'm here. Do you hear me?
Yeah, Jonas, we can hear you.
Okay, good. I hear you also.
Yeah, thank you. Just unmute. Thank you very much, Dr. Cullen, for joining us. And as many of us have seen your pre-recorded presentations entitled "From One Carrageenapite to Another: Insight in Ecomatoids from Studies on Chondrus crispus." And Professor Cullen, there are several questions that we managed to collect, and one of them is a questions that asking you to outline your experience with stress ecomatoids and the evolutions of peroxide. Is this a defensive mechanism? And if so, how does it damage the crops?
So yes, I think it's a response to any infection. A lot of the experiments I did was actually trying to see the interaction between stress and production of hydrogen peroxide. And I think it's been shown by myself and others that these stress responses are very important for the seaweeds to protect themselves. But similar to almost any organism, including us, there has to be a balance between the defense mechanism and the the capacity of the organism to tolerate their own defense. So it has been suggested, for example, that ice is possibly created by a little bit too active defense system. So in this case, ice should be produced when some kind of stress induced a too high production of peroxides, and this, instead of curing the disease or create a defense, is actually too much.
It's killing itself.
It's killing itself. So,
it is killing itself.
I tried to do experiments in Sweden with tropical eucumas, and they died. And I think the main reason was that instead of being in the sea, they were in a 10-liter flask in the lab, and then their defense mechanism was just too strong.
Thank you. Thank you, Mr. Colin, and excuse the noise from my side. And one more questions from the audience asking that from your whole organism genome work, such as with Ectocarpus and Chondrus, if informations were freely available for the ecomatoids, what do you think would be the biggest benefits to the wider community of seaweed growers and Phyconomists? Do you see these informations can help to solve issues that have been outlined at this workshop?
I would answer yes and no. In the sense that I think a lot of the information is freely available. I don't see any real use today directly to use for eukaryotes. However, I mean, we need to go further when we work with seaweeds. We really need to get better methods for strain selection. We need to be able to do crossings. We need to do plant breeding. And I think when we can do this, the availability of genomes and genome information will be an enormous advantage when we try to create better breeds of seaweeds. So today, no. In 10 years, in 15 years, yes. I can't hear you.
We can't hear you, Sudirman.
Okay, yeah, thank you. Thank you, Professor Colin. And one follow-up questions from Dr. Flower Mansuya to elaborate further of what you have said. And she informed us that she also tried culturing ecomatoids and in tanks in Israel, but they produced the peroxides that kill them. Do you have some response to Dr. Mansuya questions?
Yes, I mean, I failed to grow eucuma in the lab, but I've seen people that have done it. They have typically added quite a lot of antibiotics. And I think if you want to grow eucuma in the lab or in small volumes, you need probably to control the bacterial growth to reduce the defense mechanism. So I think well, according to the literature, and my guessing is that you need to control the bacterial growth in your cultivations.
Okay, yeah, thank you for the elaborations. And one more questions before we move to the next panelist, Dr. Simon Davis. One participant asked that he or she found in your slide on carrageenan biochemistry, and it appears that the cells can naturally convert lambda carrageenan into kappa carrageenan. Conventionally, this is done using alkali. Could this be replaced using enzyme, for example, in the future?
I mean, there is a willingness in industry and to use more green or blue technologies to do chemical reaction to produce your product. Today, I doubt very much that this would be economical. In the future, I don't know. Treatment with alkali is relatively inexpensive. So maybe in the future, probably not today, probably not in the near future.
But there are some possibilities.
There are, I think it could be done.
Okay, yeah. Okay, thank you once again, Professor Colin. And perhaps you are going back to you if time allowed after facilitating time to our next panelists. And now we are going to discuss with Dr. Simon Davis. Dr. Davis.
Hello, Dr. Sudirman. It's wonderful to be here.
Thank you. Thank you. And with Dr. Inaya, we and the team here in Makassar, Hasanuddin University, we also collect some questions specifically asked for you. And one of them is about your comment on the longevity of carbon in the system as captured by cultivated seaweed. What would be the end use of the biomass if used to mop up nitrogen, presumably from anthropogenic sources? And this is from, again, from one questions from Dr. Alan.
Thank you. Nitrogen and carbon are really two different aspects to look at in terms of using seaweed to capture nitrogen and carbon. Starting with carbon, because I think there's a lot more known about it and it's a lot bigger push. There's three mechanisms discussed so far. The first of seaweed sloughing off its skin, so to speak, as part of its growing process, and that could be classified as carbon captured. The second being in the process of farming, small pieces of seaweed could be fallen off as detritus, and that's absorbed into the sediment bed. And then the last is models of open ocean seaweed farming where biomass is sunk to the bottom of the ocean, below 1,000 meters, where it's in theory sequestered. And at this stage, it's theory. But there's increasing work in this area, particularly by there's a project led by Oceans 2050, which was a study done with seaweed farms around the world, and there should be data published on that later this year. That's regarding carbon sequestered by seaweed farms. So that's really, really interesting. That will be a good first step, and I expect that there'll be more studies following that. And the hope is eventually we can have existing seaweed farms that, as well as growing seaweed, we can also classify them as carbon sequesters, which would be a wonderful opportunity for seaweed farming communities to earn extra revenue whilst doing the same activities. But as I said, the first step from that will come in later this year, and then I feel it will grow after that. In terms of nitrogen, well, that's a lot, further behind in the development stage, and that's predominantly because there's more push behind carbon politically. There's been a first step in Australia about having what's called nitrogen credits. Having for the Great Barrier Reef, there's a credit scheme where nitrogen polluters, say for example, aquaculture polluters or farmers who over-fertilize their land and can offset their nitrogen. It would be wonderful to see that come into the coral triangle, because there's no doubt there is a significant amount of nitrogen pollution. However, I think due to the lack of political will, there's a problem of enforcement, and I foresee that's going to take a lot longer to gather momentum. So in terms of nitrogen, I think we've still got a lot further to go, but it's definitely possible. But carbon's very much on the horizon.
Okay, yeah, thank you. And some questions they could be still related to the previous questions asked by one of our colleagues from Pakistan, and Ghazalabad. And he or she mentions that in your presentations, you show that alga species such as ulva and caulerpa are the future species in this sector. Yes, perhaps this is there's a growing species also grow on the rock and rapidly in Pakistan, but actually it is neglected site in this particular country. Could you suggest how people in Pakistan can convince the public that these are sea vegetables and that we can use them in our daily life or to feed our animals? And as people there try to use sargassum as fertilizers of the growth of wheat and got good result, but having no source to make a pilot plan. How do you think these possibilities can be enhanced?
Unfortunately, I haven't seen the phycology scene in Pakistan, and I haven't certainly been to the coastline and seen what's growing, so I can't entirely comment with confidence. What I would just encourage them is to look at what models have worked in other countries. So with caulerpa, it's traditionally used in the Southeast Asia region as a food, as a salad. With ulva, there's been uses as animal feed supplements, as fertilizers as well, also in bioremediation of aquaculture. Sargassum is a bit more tricky in terms of monetizing that. But there are examples, from other uses of sargassum that, from countries like Indonesia, and then growing focus in the Caribbean with their huge sargassum issues. So I would encourage them to look at examples from other countries that are already using these particular species. But in more of a long-term aspect, there's opportunities in these particularly new species, ulva, caulerpa, looking for high-value compounds. And that's more of a biotech play. It's going to take time. And there's opportunities in that as well, but it's more of a longer term, high investment project.
Okay, thank you. And one more questions from Dr. Nesh. He mentions that at the moment there is tremendous buzz about seaweed, and there are many meetings, but it seems to him that there is no way to speed up the long slog of R&D and testing and certifications and miasma of red tape that requires for so many likely seaweed applications. What are your thoughts on how such rapid development can be materialized?
Yeah, I completely empathize with Ian's feelings about the potential length of time this could take, but I see it that if you look at the trajectory of previous development of, say, the last 2 decades in tropical seaweeds, and if we continue that trajectory, yes, we are looking at another 1 or 2 decades. But I would also point out that in the last particularly 5 years, there's been a lot more innovation and development in this space. And You know, I see that we're approaching a tipping point where, you know, where innovation will speed up a lot faster, and it will trickle down into, you know, real in-farm advances. I see Capophycus, you know, emerging from the box of being purely for hydrocolloids, which it has been for the last, you know, 40, 50 years. And it's now, you know, breaking into new markets. And that will be the big pull as market demand pulls greater demand and greater innovation in the field by using Capophycus and other euchematoids in different uses. Whether it be, you know, feed, whether it's bioplastics, fuel, you know, all the different uses. But of course, you know, there still is a huge burden of R&D that needs to happen. And the question also is, where will this come from? You know, I think the biggest challenge is the countries where euchematoid farming takes place, you know, Indonesia, Philippines, Malaysia, Tanzania. There isn't an abundance of government funding to fund the R&D. It's very different to, say, the scene in Europe or North America, where there's a lot more funding but a lot less farming. So there's a lot of challenges for, you know, how this funding should take place. I see, you know, obviously we really hope that we can get funding, but we should not wait. We can't just wait for that to happen. Because we could be waiting a long time. I see opportunities for new entrepreneurial companies moving into this space, and that's already happening now, and that will continue to snowball. And lastly, I see opportunities for existing processing companies to be more intimately linked with farmers. At the moment, there's a great disconnect between processors and farmers. You know, it's very opaque. And that means that processors have got next to zero incentive to invest in innovations in seaweed farming. And so there's very little innovation trickling down to the farmers themselves, because the processors, the buyers, don't have an incentive for an improvement. With increasing deeper relationships between farmers and processors, there will be a greater incentive. Now, key to both of this, both the entrepreneurial companies and processors investing, comes down to protection of IP. And that is definitely an issue. If there's going to be any sort of investment from industry companies, there needs to be greater protection of IP in the countries in which we operate, because otherwise they won't invest. And that is absolutely critical. And I see that as a problem that needs to be addressed.
Yeah. Yeah, one request for you, Dr. Simon, from Dr. Alan, to provide a link in relations to the carbon sequestration report that you had just mentioned. It's going to be very useful if you can provide a link about that report.
Yeah, so as I said, the data will be out later this year, so we don't have a report at the moment. I'd encourage people to look at the work of Carlos Duarte.
Okay.
But I can share a link to the website of the group. Happy to do that.
That's great. And one last question before we move to the next panelist. On the more optimistic side, one of our participants asked if once everything is in place, how many new species or cultivars of tropical seaweeds do you think could be brought into Phyconomy practice? And what are the most attractive present and future markets to produce the new biomass for?
Wow, that's a wonderful question. And honestly, I've got no idea. I think there's going to be tremendous diversity needed. Particularly as we start to innovate in new product uses for euchematoids beyond hydrocolloids. And particularly as we start to dive deeper into gene-environmental interaction. And there's no doubt that certain varieties will perform better in different locations or different seasons. But there needs to be a lot more research into this area. And from that research will come greater opportunities in different cultivars.
Thank you very much once again. And if time allows, we still have several questions for you after facilitating discussions with other panelists. Thank you once again. And now we are moving to have discussions with Professor Paik Im Lim. And many of us had watched the pre-recorded presentations on the overview of molecular markers for identifications, taxonomy, and phylogenetic study of ecomatoids. Professor Lim, we also have several questions from the audience for you. One of them is asking about the possibility to identify disease resistance through molecular markers. Can these techniques be employed, and how do you think the prospect of these techniques?
Okay, thank you, Professor Sudirman, for chairing the session and also for the questions. Okay, so as regards about using molecular marker to identify disease resistance, I would say at this stage, we do not have enough data or information to design a molecular marker that will be able to help to identify with regards about disease resistance. So further research need to be carried out. Firstly, we have to identify what are the genes related to disease resistance that we want to look into, probably what are the key genes, because like, for example, for eukaryotes, when we talk about disease, we always talk about ice. So what are the key gene that will be expressed if they are infected by ice, or we can have a early warning system. So we need to have further R&D before we can design molecular marker to allow quick detection or early detection.
Yeah, and one related questions asked by our participants in relations to the transferability of the technology or methods. Do you think these can be relatively easily transferred into other countries with poorer or more limited facilities?
Okay, thank you for the questions. Okay, I would say that molecular identification is something relatively simple technology that you can easily transfer, and also the capacity building can be built out. So I mean, for example, through Global Seaweed Star program, our institute, we have actually accepting students from Mataram University to come into laboratory, and we teach them how to use this molecular tool for identification. But at the same time, I also understand, because some of the cultivation farm is located as quite, especially in Southeast Asia, some are at very rural area that access, even sometimes basic electricity might be a problem. So I mean, in order to help the industry, of course, a province or national level center of excellence will help to build up, but as well as local university. But the thing is it's not a difficult technology. Yeah, it's possible to be built up. Yeah.
Yeah, that's great to hear. The possibility for technology transfer. And one question particularly addressing Malaysian context. Many of us know that Malaysia's seaweed industry went into a period of declining. And based on the molecular work you are currently doing, what do you think are the best strategies to recover or to revive and in fact can be valuable to increase productions in the country?
Okay, thank you for the questions. Okay, so I would say that when we talk about the decline production in as regards about seaweed in Malaysia, there are many factors or several reasons that I won't say that molecular marker or molecular methods will be able to solve the whole problem, because we know there's a few problems. I mean, we know that when you increase the cultivation area, you can increase the production. But another things that I think that when we talk about molecular ID that can help is actually using molecular tool, it can help to be used for the selection of the best seedling that can be used for growing, such as fast grow rate, pest, and pathogen free. Because if you do to look into, because in year 2014, our group published a paper that's on the genetic diversity of the euchematode in the Southeast Asia country. And based on the study, it shows that the three main species that we are cultivating for the region, like Kappa ficus avrazia, Kappa ficus stratus, and Euchema denticulatum, is very, very low as regards about genetic diversity. So if you were to trace back on the history of domestication of euchematode, we know that seedlings are being transferred out from Philippines or Southeast Asia region to different parts. If you were to look to the time of the size of the Kappa ficus avrazia, for example, is very huge compared to currently what we have. And the main reason is we depend on only vegetative cultivation. We know that if we are using the same mother stock that continue to vegetatively culture, so with time, the quality will deteriorate. So we need to find, through molecular tools, to identify and to find from the wild population, as well as to look into the genetic diversity. In that case, we can introduce more diversified seedlings for cultivation, and this will be healthy. So, and also less susceptible to any outbreak of disease. Okay, so I hope this will somehow to help. So another potential aspect that was not included in my talk is using transcriptomic to help to identify relevant genes related to stress. We know that temperature increase have been one of the, with the global climate change, is one of the challenge. So our laboratory, we are studying on the identify the strain of species that can adapt to higher temperature. So at the same time, we also use transcriptomic tool to identify what are the relevant genes that's been identified. So these genes are potentially can be used as a probe, but this will take some time before we can share the result of find. And I think if there's a concerted effort among the country to pull out the strength and to look into different aspects, I think that will greatly help the industry. Okay, thank you.
Yeah. Thank you, Professor Lim. And one last question related to the potential for future commercial aspect. And this participant asked if new species should be considered, and can molecular markers can help your ID species of interest that can become commercial success in the future?
Okay, thank you for the question. So I would say that molecular techniques or molecular ID is a tool that assists us to confirm as regards about the genetic composition. So as we know that seaweed are very plastic in nature, the morphology are quite easily changed according to the environment. And we can see already in Kappa ficus and Euchema species that when we move them into different environment, their color might change, their morphology might change. So in this case, the molecular marker is needed. And then, and for even for when we describing the new species, or to identify whether this species is the same species that we have planted after many, many decades, or this is a new, we need a tool to we need molecular ID to help to confirm whether they are new or old species, existing species. So, and as I mentioned previous question, molecular ID will help to assist the best seedling that potentially can be cultured, and that will indirectly contribute to the success of commercialization. It won't be directly contribute. Okay, so this is what I think. Okay, thank you for the question.
Yeah. Thank you very much, Professor Lim, for the further elaborations from the questions asked by our audience. And now from Malaysia, we are going to move to the Philippines with Dr. Michael Roleda. Dr. Roleda, can you hear us?
Yes, I could hear you. Good afternoon. I was muted.
Okay, good afternoon.
So, yes.
Yeah, your presentations on wild Capapicus populations and selective breeding as sources of new cultivars also attract numerous questions, and I would like to choose some of them and ask your further elaborations. One of the questions is if it is indeed your presentation is indeed very informative and with considerable potential to improve the cultivars of ecomatoids, whether the strains selected for breeding were homozygous for specific trait. If so, what is the trait? This is a question asked by one of our participants. Could you please respond to that?
Okay, thank you for that question. So the trait that we are selecting is basically a high growth rate, and that is our preliminary determinant. And then when we observe a high growing novel cultivar, then we also try the biomass and let our collaborator analyze the carotene yield and quality. So in the hatchery, we first select the highest growing cultivar, and then thereafter it's the time that we look into the chemistry. Thank you.
Yeah, thank you for the clarifications, Dr. Roleda. And questions from participants, Kelvin Zhou Kang Ong, asking about how does the inductions of germinations and sporulations of the seaweed is being done, and how long does a seaweed take to finish its life cycle?
Okay, that's a very good question and might take a long time to answer. Okay, first, we could induce spore release. So spore release could either be induced by desiccating the sample for a certain period of time and then put them back in water. But in our case, we do the natural sporulation technique. No? So what we did is just cut the piece of the thallus that there is the cystocarp, or when it's tetrasporic, the piece of the thallus that we see the tetrasporangial nematotia. Cut it in 1 centimeter length, and then disinfect it, and then just put it on top of a glass slide, and we wait like a couple of days to let the spore naturally release. So we look at it regularly under the microscope if the spore is released, and then we take off the segment and then put it in another container to let it continue to release. And then it took about like 5 months rearing them in the lab condition. And then after 5 months, we brought it into the hatchery condition. And to complete the life cycle, the F1 generation, that is not yet a complete life cycle, that is from tetrasporophyte to gametophyte, that the gametophyte becomes fertile, that was about 10 to 11 months. And then the F1 generation, we also induced them to release, and then we are completing that life cycle now. So the next generation is now in the lab and waiting to be transferred to the hatchery. Yeah. So
thanks for the explanations. Yeah, and one more questions from our audience asking about what conditions are missing in the most commonly cultivars strains, which disable them from either sexual or asexual reproduction through sporogenesis. And is it their reproductive structures are entirely absent, or do they sometimes form during cultivations?
Okay, so that's an interesting question, no? Because most of the cultivated cultivars now, we don't usually see reproduction, whether they are gametophytes or tetrasporophytes, that we do not know. But there was a report from De Paula that they imported seaweed from the Philippines, and it turned out to be tetrasporophyte, and then they were able to induce tetraspore release and then grow them until a certain stage. So whether what we are cultivating now is tetrasporophyte, and whether there are farmers looking into the cross section, because it's not easy to see the tetrasporangal nematocia, no? Or whether there are scientists continually doing some sampling and doing some cross section to look at the tetrasporangia, I do not know. So because it was easy for us to detect reproductive structure in the hatchery condition. And in the hatchery condition, we're just using 70-liter aquaria, growing the progeny there. And after 7 months, they become fertile. So we actually have a male gametophyte, a female gametophyte, and a tetrasporophyte in separate tanks, because eventually we separate them, each individual, each life stage, we separate them into different tanks. And then we see reproductive structures. How come we don't see this in the field? I do not know. Maybe we need to do more observation, or maybe because the plant in the field is continuously asexually propagated for maximum about 60 days, and within 60 days, they might not be able to produce the reproductive structure. Like, for example, in our case, it took about 7 months before we were able to see reproductive structures. So if we could have a setup in the field, cultivated strain, and then just grow them to 7 months without harvesting, I do not know if we will be able to find some reproductive structures, if we will be able to establish if the farm cultivar is a tetrasporophyte or a gametophyte.
Thank you very much for the valuable insights, Dr. Roleda, and also for the detailed elaborations of the questions. And now from the Philippines, you are going to move to Australia to discuss with Dr. Zanny Langford.
Okay, thank you very much.
It is great to meet you. Thank you, Dr. Roleda. Dr. Langford.
Hi.
Zanny. Hi, yes. It's great to meet you here.
Yeah, hi, good to see you.
Yeah, and the title of the presentations is Remote Sensing of Ecomatoid Productions. And like other panelists, we managed to collect some questions related to your pre-recorded presentations. One of them is, will you be able to use multispectral cameras and scan for the disease and or for poor growth?
So this paper was a collaboration which was part of our previous work that we'd done using Planet Labs imagery, with some new work that we're doing with some of our colleagues looking at automating that process and other sources of imagery. So my co-author for that, Swanti, is also on the line. So she's better placed to speak about the technical aspects of the process, whereas I'm more looking at the social and economic aspects. So are you there, Swandi?
I'm here. Hello. Yeah, so multispectral. So I was looking at using satellite imagery, and what I found actually was that using NDVI, which is a vegetation index, so which is basically a ratio of the red and near infrared band. Actually, using that, I was able to do quite good segmentation of seaweed beds. And so, yes, definitely multispectral cameras, I think, is very useful to doing the segmentation task. I don't really know much about seaweed, so I don't really know like any sort of physiological aspects to this, but just from the imagery point of view, the multispectral is important.
Okay, yeah, thank you. And one more question that perhaps you both can elaborate. Our participant asked how much more detail can you get or can you obtain in discerning the crops?
In the what, sorry?
Detail. How much detail that you can get?
Well, it's limited to the scale of the imagery, so not a lot of detail. It's sort of good enough to get an approximate idea of the scale of production and how that varies between seasons and between years. Mm-hmm. But it's definitely not at the same level that you would get from drone imagery. So using this data, I guess it's really important to combine it with other sources of information and data collected on the ground and other types of data if we're looking to get better accuracy, but also to make sure that what we're sort of seeing is appropriate and to sort of ground truth the satellite analysis that we're doing.
If I can just jump in there. Okay. The satellite images are, so Sentinel-2 is on a 5-day basis, so we can actually get data on a weekly basis as long as the cloud cover is, as long as there's not so much cloud cover. And there's also planet data, which Zani was looking at on a monthly level, but they also have just daily images, which are at a 3 meter resolution. So we can use that as well to get data at a daily level. But what I found was that earlier in the season, earlier in the year, there's a lot of cloud cover. So more work has to be done on doing cloud masking to get data from the early months.
Yeah, and thank you. One more questions is, what do you see as the future of imaging related to Phyconomy in determining seaweed growth and health?
I think that there's quite a lot of opportunity for using that data. I think at the moment, the data, at least in Indonesia, that's produced officially is not very accurate. So I think there is a lot of scope to improve the accuracy of data collection. I think that could have wide-ranging implications for a range of different actors in the value chain to help improve coordination of the industry. And it can help with planning all sorts of things from distribution of seeds to drying or collecting, a better understanding of where seaweed is being produced and when could, I think, be used by a large number of people.
Okay, thank you very much, Dr. Zani, for this. And due to the time considerations, we would like to move to the next panelist, Dr. Anisha Hurtado and Dr. Alan Critchley. And we also have several questions from the audience.
Yeah,
Dr. Anisha and Dr. Alan, yes?
I'm here.
Okay. Yes, thank you. The first question is, what do you think the key modes of actions might be? Have these been investigated to the same levels as impact of phycostimulants on land plants? And if not, what needs to be done? And are there any collaborators in the audience who might be interested in this aspect?
A very interesting question. In fact, I really prepared that the response for this because firstly, seaweed extract biostimulant has been used for how many decades in agricultural crops, and it's only lately in 2009 that it's being used in seaweed cultivation, primarily for cumatoids. But now we have other seaweeds. So the first thing that we were looking into is, of course, the acceleration or hastening of the growth rate, and then will this have an effect on the incidences or the occurrences of disease and pest infestations? So with our several experiments in my team, not only in the Philippines, but also in Malaysia and also Brazil, we have found out that using the seaweed extract biostimulant has really accelerated growth rate and reduced the incidence of ice in particular, and even epiphytes and the phytes. Now the question is, what is the mode or pathway of this seaweed extract in terms of the efficacy on the reduction of diseases and pests? That is the only missing link now to our studies. So I have been looking for partners to work with me and to work with Alan and me also, and the rest of the team in Southeast Asia, especially Malaysia, to this effect. And I welcome collaborations, and I'm open for it.
That's great to hear, Dr. Anisha. And at the more technical level, one participants asked he found that it appears micropropagations result in good propagals, and it would be interesting to know how fast the scale up is, especially with AMPEP. How much time do you think would it take for a kilo of seaweed to yield, for example, 100 kilos of microphagal material?
Yeah, again, and a very interesting question. And I was really challenged because based again from our personal experiences with my team here in the Philippines and in other countries, it takes around, okay, let me give you a very short span for the whole propagation, that it takes only 30 to 45 days in the lob, and then we bring them in the land-based hatchery. If in case the weather in the sea doesn't warrant cultivation, so we keep them for the transition period, and then after that, another 30 to 45 days, we bring them to the sea. And then afterwards, 30 days, and afterwards, wow, we will be going there for the line cultivation. So practically, we can do it in 4 to 5 months. Ready for, we have already cultivars, or rather seedlings for cultivation. And your question is how many days will it take 1 kilogram to reach 100 kilos? Okay, so I base this answer from the growth rate, that assuming that you will maintain a 7.8% per day of growth rate, then 1 kilogram will reach 100 kilograms in 90 days, equivalent to 2 growth cycles of 45 days each.
Thank you, Dr. Anisha. And one last question for you and Dr. Alan. Our participants said and asked, the AMPEP is derived from the brown seaweed as Ascophyllum. What is your opinion of extracts made from other seaweeds? How do you feel about this?
Yeah, of course, if the participant listened to my concluding remarks in my presentation, it is not only Ascophyllum nodosum that can be sourced as a seaweed extract. We have other brown seaweeds in the temperate countries like Eclonia, Macrocystis, Saccharina ondaria, and in the tropical countries, we have the Sargassum, we have the Turbinaria. However, these are not yet tried commercially, or probably they have been tried, but not that into a big scale. So there's no harm. There's no harm in trying other sources of seaweeds. Actually, when I first used this Ascophyllum, I studied the amino acid content of this Ascophyllum seaweed extract powder, and I found that it has a lot of amino acids. And I tried it first in 2008 on the micropropagation, because I have been previously, since 2000, I have been using the PES, the SS over 2, or even the Von Stotts as my culture medium for tissue culture, but those are really very expensive. So I tried using this seaweed extract from Ascophyllum plus PGR only, and it worked. I was able to generate direct axis plants of Capophycus of different strains already. So we published that paper, and now other countries are following. So it's not that Ascophyllum is the only source of seaweed extract for micropropagation. There are a lot of brown seaweeds, perhaps even the red seaweeds or the green seaweeds, to be tapped and explored for purposes of micropropagation.
That seems promising. And thank you very much, Dr. Anisha. There are still many questions actually for you both, but due to limited time, we are going to invite our next panelist, Dr. Nesh. And
okay,
Dr. Nesh's presentations, thank you, is the seaweed industry development and lesson learned from Indonesia to the world. And there are several questions already offered by our participants. And one of them is in relations to the stakeholders. The world, of course, need more seaweeds, not only for the ecosystem function and food, but also to help to complete the United Nations SDGs. You have provided valuable insights in your presentations into what next. How do you think these stakeholders and producers are they listening, or will they listen? And if not, could you please predict what are the good, the bad, and ugly scenarios for the future? Dr. Nesh.
Okay. Well, number one, before I answer that, let me say that our company, C6, is making extracts like Ann was just talking about from Capophycus. So we have a factory in Bali that is making that, and it's already an established product. Okay, to answer the question you just asked, I think I addressed this in the section about value chain governance. And to me, the way forward is relational value chains, where all levels of the value chain are collaborating with each other on developing new products, on implementing sort of biosecurity measures and all the rest of it. In other words, under a relational value chain governance, all value chain levels are talking to each other and collaborating with each other. That is the key. That pulls in all stakeholders.
And one question was also raised in relations to lesson learned in Indonesian region. As we all know, Indonesia is a large country with many different islands and different circumstances. How do you think the similar development can be expected to be implemented or scale up in other regions within Indonesia or even within Southeast Asia region?
Well, I think lesson 9 had to do with the coral triangle. And I think that, you know, there is just massive development potential in the whole coral triangle. In Indonesia, we are blessed because we do not have typhoons and we do not have wars going on. Those are two things that make it possible for people to go anywhere in Indonesia and connect value chains. As you go further east to Papua New Guinea or even into Papua, Indonesia, Solomon Islands and so forth, I think there's huge undiscovered potential. And even in East Indonesia, I have lived in East Indonesia, in Maluku, and in Ambon, and in Tuau. And there's just a mind-boggling opportunity in that whole area. So Indonesia, in particular, the Coral Triangle in general, in my opinion, has the capacity to increase seaweed production by an order of magnitude over what it is now.
Thank you, Dr. Nesh, for these valuable insights and also for answering the questions. There are still many questions for you actually, but unfortunately, we are now preparing for the last sessions. So I would like to provide a brief summary to our sessions. I think we have learned and discussed several good practices from several countries in Southeast Asia and also Australia and Latin America, such as Chile, and also some latest development offered by our colleagues from UK and France. And we can see some opportunities for further collaborations to further develop this tropical Phyconomy. And we are very pleased to enjoy both of your pre-recorded presentations and your responses to the questions. Once again, thank you very much for your time, and we apologize to all participants who offer us some questions, but we do not have enough time to respond to their questions. And I would like to invite my colleague, Dr. Inaya, to also provide some concluding remarks. Dr. Inaya.
Thank you, Sudirman. Time on my on the screen, we still have 12 minutes to go. So I think we still have, there's at least 2 questions that come during the sessions, and I found it's very interesting. Maybe we can go for that if it's okay. We still have 12
yes, please.
to go. Okay. Yeah. Can I just go for it? The question is go to Dr. Anicia and for Simon. This is from India. I found some Capophycus species tissue culture didn't growing well in some production areas. It's due to the transition of season from rainy to dry season. It's not growing well during the dry season. How to minimize the risk? The tissue culture could not grow well after 3 crop cycles. How do you think, Dr. Anne Hurtado and Simon Davis? Go for Dr. Anicia first, and then later on Simon. If not, I think we still have time for that.
Okay, I'll go for the last question, or I would like to react on that. The tissue culture, the Capophycus is only good for 3 growth cycles. Yes, I agree with you, because we have already some experiences in the Philippines, especially at Seaf Deck, where we outplanted some in a seaweed farm here in Iloilo, and after 3 crops, they didn't grow as much as the first and the second growth cycles. So I think that's one of the advantages of having the land-based nursery, wherein you have to while the temperature in the sea is not favorable for field cultivation, then you have to keep your seaweeds in the tank so that you will be expecting positive results if you bring them out, because it's not enough that if you have already some plantlets in the lab or in the land-based hatchery that you have to bring them out. You have to synchronize it with the environmental conditions of the sea, because for one thing sure, seaweeds grow much faster in the open sea than in the lab. So you have to be your weather conditions should be synchronized with your programming in the production of your plantlets, because if you don't do this, you will just be having an exercise in futility, and your plantlets will just put into waste. So you have to make a program on how to operate from the lab to the sea, to the land base, and to the sea base, or even into the field cultivation. Otherwise, and you have to really synchronize it with your local weather conditions to minimize high risk.
Okay, thank you, Dr. Anicia. Simon,
welcome.
Please.
Yeah, thank you very much. I think I'd like to start where there's a couple of questions that I have really before I could give a thorough answer. And the first is one that Dr. Reddy's listed in the chat is, when we talk about tissue culture, is that from callus or is that from micropropagation? Because there is a lot of confusion in conversations, and they're very, very different types of technologies. And I think I agree with Dr. Anisha, you know, environmental factors, seasons are crucial, but it would be worth zooming in a bit and discovering, is that declining superiority after 3 cycles, is that due to a certain fatigue, a declining advantage of the tissue cultured seedling? Or is it due to a mismatch in the season? It was previous in an optimum season, and then it's just in a less optimum season. So I've got more questions than answers, I'm afraid.
Simon, can I answer you with your question?
Yeah, please. Yeah, go for it.
Yeah,
go for it.
actually, I gave that example here in the Philippines for three growth cycles, which is the something like short period, but they use a different, not seaweed extract as the culture medium, but what my collaborator or my partner in Malaysia is using is purely seaweed extract and a little only of the PGRs. And until now, we started doing it in Sabas and Puana 2018. And then it still continues up to the present. So the same protocol we are using. So far, so good.
Yeah, fantastic. So that was micropropagation, correct?
Yeah, actually there are two prongs in the micropropagation using culture techniques. You have the callus, from the callus, and from the direct access formation. So gotcha. We use the direct because it's shorter in time. In a shorter period of time, you have already new plantlets, because callus are undifferentiated cells that it has to transform into differentiated cells to generate plants, the whole plant. Yeah.
Yep. Yeah, absolutely. And I think, thank you, Dr. Anisha, for this. And in our dialogues, I think we should really be more specific. Are we talking about from callus, or are we talking about from micropropagation? Because in communications, there's been a lot of confusion. And I would just suggest that for communication in the industry and academia, about being a bit more specific that way.
Okay. Thank you very much, both of you. Thank you. And we still have 6 minutes to go. Hopefully we can do effectively. There's one question for Dr. Lim. Dr. Lim, are you still here? Hello? Dr. Lim, there's one question.
Professor Lim?
Yeah, I'm here.
Yeah, Professor Lim. Okay. Yeah, there's one question. Hopefully you can answer it in 5 minutes. Are the available dendrograms or haplotype network, which include worldwide achomatoid specimens, could infer biological patterns other than taxonomic or biogeographic pattern?
Okay, thank you for the question. Actually, I already answered in the chat box, but it's okay.
Oh, okay. There's another one question. If it's, but your comment, please.
Okay, so as regards about the haplotype network, is actually just to review the genetic composition of the strain that is available. So it's not really for looking into phylogenetics or taxonomy. Yeah. I hope I answered the question. Yeah.
Okay. There's there's another one I didn't follow. Is it already answered too? It's about the sorry, what is it? It's about the can we improve the vigor of Kappa ficus through polyploidy techniques? Your comment, please. Is it already?
Can you repeat your question? I didn't get.
Yeah. Can we improve the figure of Kappa ficus through polyploidy techniques?
I think at the moment we do not have any data for euchematoid, so we do not carry out the experiment. So I think it's very hard for us to comment at this stage of time. We need experiment to look into that. Yeah.
Okay, thank you, Professor Peck. Thank you for all panelists. Thank you, everyone. We finally come to the end of the session 3. At the moment, we will now watching the video on opportunity for collaboration on seaweed safety. After the video, we hope that you can join us for the session 4 later at 4:15 in Makassar time. I think that's it for the session 3. Thank you very much for everybody, for all the panelists, for all the participants. Me as a facilitator has to say thank you and see you in session 4.
Thank you.
Thank you.
Everyone, first I would like to thank Alan Critchley, the organizer of this workshop, for inviting me to talk, but also to involve me in the practical organization of the Tropical Phyconomy Coalition, and also to provide some support from the IDEAL project to organize this first workshop. I am Philippe Potin. I am currently the scientific leader of the Safe Seaweed Coalition, together with Vincent Dumézel. And during the last 10 years, I was the scientific coordinator of the French IDEAL project. Since 2017, I started also to coordinate the European Innovation Action, GENIAL. Today, I will talk about what could be done in the context of euchematoid research to solve some issues this cultivation is facing. I started to be involved in this discussion about 20 years ago, thanks to Alan Critchley. And we discussed at that time with Anicia Hurtado, Jonas Collen, and Sylvain Faugeron about the opportunity to apply for a call for proposal from the European Union in the context of international cooperation. We named our project IDEAL at that time. Unfortunately, this project was unsuccessful for political reasons. I will try to convince you today that the science basis which were behind this project are still valid about 20 years later, and they are still conducting the future of research for around euchematoids, and they will probably provide some solutions to solve the issues this industry is facing today. Today, I will not talk too much about the Safe Seaweed Coalition because it was developed by Vincent Dumézel, but I will try to show you how this new context of for the international coalition will provide opportunities to solve some very important issues and support scientific cooperation on euchromatoids.
I'm also co-leading with Philippe Potin and a few others the Safe Seaweed Coalition, which is hosted by the CNRS. And once again, we'll come back on what it means. What it means is that we are trying to build a global seaweed community to get together and address the issue that needs to be addressed to really scale up the seaweed industry and take the most of it. And they decided to support notably the seaweed business because they think that seaweed can really be one of the most untapped resource, maybe the greatest untapped resource we have on the planet, and that if we want to address the challenge of our generation, we need to use seaweed. So Lloyd's Register Foundation decided to fund £4 million for this once again this global seaweed coalition. And we'll see what they will do with that. And supporting the Lloyd's Register Foundation, we have another organization which is very global and very international, which is the United Nations Global Compact. So a quick overview. So it's the world's largest corporate sustainability initiative. It has been brought together by United Nations, and it's a voluntary initiative based on CEO commitments from over 13,000 different businesses committed to the principles of United Nations, very sustainable, committed to the sustainable development goals that you can see on this screen. That was the first step towards the Safe Seaweed Coalition. So the manifesto was a way to tell the story to everyone. How good is seaweed? What is the potential of seaweed? This manifesto was science-based, and this manifesto was really around what is the situation, where are we going, and how can we enable the potential of seaweed. So we have thousands of downloads. It went very, very big when we launched this manifesto last year in 2020, and it started to be very, very successful. So the manifesto, you can download the manifesto here, www.seaweedmanifesto.com. The manifesto vision was the following. The seaweed industry is immature and fragmented outside of Asia. We need to support bringing people together. We need to convene the seaweed stakeholders. There's a lack of global standards as well. There's a hilly patchwork of regulation from many different countries which are not consistent at all, and we need to support more consistency in terms of standards to really enable the creation of a global market. What is this global coalition? Once again, the global coalition is funded by the Lloyd's Register Foundation for £4 million. It has £1 million is going to Wageningen University in order to really support fundamental research on safety for seaweed. £3 million to the CNRS in order to support the building of a global coalition, gathering all seaweed stakeholders and building collaboration, as well as supporting directly projects in order to improve the level of safety. So 80% of these £3 million are to be distributed by the coalition to projects in order to support safety, support standards and regulations, support innovation for safer production, ensure high-skilled operators share knowledge. Then the scope, what we have seen, improve global standards, upscale the seaweed industry with a collaborative and optimized supply chain. That is the objective of the coalition, and I would call every one of you to join this coalition, and we will see how to do that right now. Where we are gathering experts across our members, so those who are already registered, and you have the list of topics that will be discussed by a group of, we want a group of 10 to 15 people to discuss what should be the priority for the coalition. What should be the priority? Should we fund environmental safety? Should we follow global standards for environmental safety? Should we fund something, a training program? Should we focus on a certification program? Should we focus on traceability? What should we do? How to give visibility to investors who are willing to invest? So what should we do, being innovative, using at best these £3 million that were invested by the foundation? So we are we have a 2 to 3 months ongoing discussion right now. It's going on. You can register to the foundation and raise your interest to participate to one of these roundtable to say, hey, we need to fund something related to that, or there's this project which is interesting that we may need to support something in around this project or around this thing. So once again, over to you, join us, be part of the coalition. The membership is fully free. You just need to register, which is a very easy process. And once you are registered, you can become part of the global map. You see here, you have a list of, when you go into our coalition, when you go in our portal, which is the safeseaweedcoalition.org, where you can register. We'll be glad to support you. We are at the very beginning of an exciting journey, building a global community. It's a charitable work. So Safe Seaweed Coalition is led by Philippe Potin, that you know very well. We have Kevin Casella and Azedine Badis who are taking care of the coalition on a day-to-day basis, and we'll be glad to support you if you have any problem with the registration.
connected.
facilitators 4. The time is yours, please.
Okay, thank you. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Respected speaker, excellent speaker, and distinguished participant. Welcome again to the Tropical Phyconomy Coalition Development 1. Very warm welcome from Hasanuddin University, and very thank you very much for you to join this workshop. I know that many of you come from different zones, so it is hard to join this workshop this afternoon. For example, in US, it is early morning now, and in Makassar itself, in Indonesia, it is coffee time in the afternoon. But in Australia, we have a lot late afternoon and so on. So we are very appreciative with the hard time that you have time to join this workshop. That we have for this workshop, it is important session, even though it is the last one, because we will use this session to discuss about how to rate and how to build a partnership. For example, if you want to build a global civic community, and we need a kind of collaboration among scientists, something like that. Yeah. So we will use the information, we will use the data, we will use the lesson learned that from the previous 3 sessions that we already see before. And then it is time to discuss all these things to build a kind of partnership later on. Hopefully that we can build a very productive partnership. So today this session we have 11 speakers. So this speaker will be later on introduced by Dr. Nana Saleh, my co-facilitator in this session. But before we start with the introduction of the speaker, let me remind you again that that in this session only dedicated to the discussion and also question and answer, because we believe that all of you already watched the video, the talk of all of the speaker, and then this session we only use just for discussion. And another thing, organization committee already received a number of questions that have been posted to the forum. So me and Dr. Nana will pick up some of the questions to be addressed by the speaker, maybe 1 or 2 questions. If you had more time, and then we will have raise more questions with the with the another question that we got from the box. It is also possible to post question or comment in the Q&A box, in addition to the question that you already have in the box forum. So there are 2 ways. So we will ask question from that we got from the forum, and another possibility also that the audience or participant can also post question or comment in the Q&A box. Okay, please, Dr. Nana Saleh, to introduce our speaker in this session 4. So your time is yours, Dr. Nana, please.
Thank you. Can we get the screen out? Okay, thank you, Pak Mahatma. Ladies and gentlemen, I'm Nana Saleh, and I will be co-facilitating this session. And allow me to introduce our speakers in this session of the workshop. Can we have the screen, please? Okay. So okay, just quickly, I think most of you have seen all of the recorded video. Can we go next, please? And this is the last session of our workshop for today. Next. Okay, let me introduce our first speaker, Dr. Van Sang Domeze. He is a senior advisor at the United Nations Global Compact on Oceans and director of the food program for the Lloyd Register Foundation. And he's also the leader of the Seaweed Manifesto, partnering with UNFIO and the World Bank and WWF, universities and NGOs and large brands in a call to scale up the seaweed industry in order to address some of the world's most important challenges. And he's co-leading the first international platform for seaweed stakeholders, the Safe Seaweed Coalition on the expert panel to define the European Commission ANCAR strategy and support preparation of the UN Food System Summit. And I received a request from the organizer. We know that Dr. Vincent has to go, so I might stop here and continue later. Pak Mahatma, can we ask him his questions? Dr. Vincent, because he just have very short time. So I will continue to introduce the rest of the speakers, but maybe we can begin with Dr. Vincent. Sorry, I keep saying Vincent or Fanzang. This is very French.
Vincent.
Vincent, yes. Dr. Mahatma, do you want to lead the question for Dr. Vincent?
Okay, sure.
See you later. Thank you.
Okay, thank you, Dr. Nana. Dr. Vincent, you are the one that need to answer the question because you had limited time in this session. So, okay, I start. You clearly love all the thin seaweed. What attract you to help this cause of seaweed and their benefit for the world? Well, there are many, as we have mentioned. And first of all, I'd like to thank you very much for accommodating this timing. It's a lot of uncertainties. I'd like to mention that we are, this coalition, we are co-leading that with Philippe Potin, who is attending also today and who is part of the speakers. So he will be more than able and even much better than me to answer any question you may have regarding this initiative or more general. But no, what I would like to state here is from a UN perspective, because at the first time, I think the UN are moving into a real program and are building a momentum around seaweed, because seaweed indeed ticks a lot of boxes when it comes to addressing some of the most pressing SDGs, the Sustainable Development Goal. So food and security, of course, SDG 2, there's a critical potential as direct food for human or as indirect food supporting agriculture, like feeding animal and being a biostimulant for plants and protecting them. So there's a great potential for seaweed to support that, and we have to scale it up. And there's a great work to do in terms of pushing this forward and training and educating people to eat more seaweed or to use seaweed in their supply chain, in their farming practices. There's a great work of education, and I think that's why we need to get together through that coalition, which is the first ever global coalition. You can all register there. That's for free. I will share the link right after my talk, but you can all go to the coalition, become a member, it's important, and then you become part of the coalition. And then the coalition has been funded by the Lloyd Register Foundation for £4 million, so 80% of this will be dedicated to support projects. We will issue the proposal very soon, and the UN is fully part of it because, as mentioned, it can support SDG 2, reduce hunger, can also support the SDG 5, which is the gender equity. We see that there's a growing potential for seaweed to give revenues to poor people in coastal community and also to women. We see a lot of people involved in the seaweed business are women for many reasons. I was at another panel yesterday afternoon, and I was the only man with 8 women next to me. So that was very positive. I think there's a great potential here. There's even more potential in terms of carbon sequestration. That's something that we are working heavily on at the moment with UN Global Compact, preparing the COP26 very soon. And there's a potential there that seaweed can mitigate climate change, either by displacing some products that are emitting a lot of greenhouse gas at the moment, like plastics, like fertilizers, and so forth, or improving digestion of the livestock. We've seen some possibilities that are still to be fully confirmed from a scientific point of view, but that seaweed may have a potential to cut methane emission, which is one of the largest contributors in the world to greenhouse gas emission. So, and last but certainly not least, there's a potential for seaweed to sequester carbon down in the ocean, shedding cells when it grows. Seaweed can represent a very interesting carbon sink and the only nature-based solution to actively take carbon back from the atmosphere to the ocean sediment where it used to be for millennia. So there's a very important point here. There's a very important point about creating, I would say, a regenerative aquaculture, and that's important as well. Life underwater, SDG 14. We have to restore abundance in the ocean. We have to clean the ocean. We have to create ecosystem at sea and develop aquaculture, not only seaweed, but feed the world with a global aquaculture. We are positioning this very high in the UN Food System Summit that is taking place in Rome this summer and then in New York at fall. It's a very important meeting and summit because it's the first of its kind, but certainly not the least. And Blue Food has a slot and is seen as a game-changing solution to feed the world of tomorrow. And seaweed is fully part of the Blue Food solution because that's the lowest trophic level there. So that's absolutely critical. So there's a lot of benefit, and we can mention medicines, bioplastics, pollution, mitigating pollution. So seaweed may well be the greatest untapped resource. I'm here to voice that the UN has identified this as a game-changing solution to meet some of the generational challenges of our world, and that we have to scale it up. We have to put the right regulation, the right standards. We have to make sure it works. We have to educate people, and we have to train smallholders to become seaweed farmers. So there's a long way to go, but we will need to be all together and work together to get the funding to maximize benefit from research, from investment, to avoid overlaps, and to make sure that we all work in collaboration. The Safe Seaweed Coalition is a new home for all of those who want to work all together in this seaweed industry and really make a difference. Okay, thank you, Vincent. Before I come back to Dr. Nana, there is another question if you still have time to answer. Hey, go ahead, to respond. No problem. Okay, one more question. This is important because it is about money. So you are seen as the man with the money. Please just outline how the funds available are to be administrated. Okay, well, that's a good question. The foundation, the Lloyd's Register Foundation, which is a British charity, has granted £4 million to the CNRS. And once again, my very esteemed colleague Philippe Potin here is leading this initiative. We will have a call for proposal that will be issued at the end of this month on the 26th of July. The first one. So I think we will have a relatively small project. We will select small budget in order to bring the family together, I would say, in order to create because if we fund only one big project, then that will not have a strong convening power. And to start with, even though we already have 400 members in this coalition, we want to make sure that everyone is coming together. I shared the link, by the way, in the chat box about the coalition to join us. So you feel free to become a member and to display your solution, what you're doing in what we call the greenhouse, the Ubuntu platform, where you can tell, okay, I'm producing this, I'm processing this, I'm doing this, and then you can become a part of the ecosystem and visible to investors, visible to the coalition, visible to other stakeholders. So back on the topic of the funding and the money, we will release the first contract at the end of this year. There will be 3 years at least. So every year we will launch call for proposal. Then with the steering committee, which is gathering FAO, WWF, the government of America, of Australia, representative from Japan, from EU, and so forth, we will select and award the funding, and then we sign the contract. But there will be a call for proposal official co-proposal for that. But yeah, I mean that's the process that we have. So end of July, official communication through the coalition that the co-proposal are launched. 2 months for people to reply, and then yeah, we'll see what then there will be another one next year. And in the meantime, we will organize discussion. We have organized discussion. We have organized roundtable discussion as part of the coalition. So we have gathered a group of 15 people across our members to discuss some topical issue like biopackaging, seaweed in Africa, residues, nutritional standards, and so forth. And this is steering also the different calls that we will issue. So that's how it works actually. Okay, thank you very much, Vision, for the talk. Very interesting talk. Yeah, we hope that we have a nice time, and we hope that from this workshop we can build a global seaweed community later on. Hopefully. Thank you again, yeah. Vincent. Yeah. So please, Dr. Nana. Yeah, please, Dr. Nana, continue to introduce our rest of the speaker this session.
Okay. Thank you, Dr. Mahatma. Can you hear me?
Yes, yes.
Yes, okay.
I do.
So there are 11 panelists, 11 speakers for this session, for our session. I would like to introduce Dr. Kasmiati. She is the head of Center of Excellence for Development and Utilization of Seaweed, and her background is food science and technology, with special interest in developing health-related and consumer products. Dr. Nick Paul from Australia, joining us from Australia. He's a marine scientist at the University of Sunshine Coast and led a project funded by Australian Centre for International Agricultural Research. And Nick plays role combining research expertise with a strong focus on industry partnerships, innovation, and IP development and commercialization. Our next other speakers from Philippines, Mr. JP Paisan. He's an associate researcher and commodity team leader for seaweed and at CFGEC-IQD, and he's an early career researcher for the Global Seaweed Star program, safeguarding the seaweed aquaculture industry of the developing countries. And he focused on disease and pest detection in eucalyptus seaweed farms in the Philippines. Dr. Cecilia Kembi, she's joining us from University of Malaya, Malaysia. And she's also a researcher in Global Seaweed Start, focusing on biosecurity and policy for seaweed aquaculture industry, in particular of the eucalyptus. Dr. G. Grace Suya, she's also a researcher at the Global Seaweed Star project funded by the UK, and she focus on seaweed value chain, emphasize on governance, social network risk, and risk management and gender. Dr. Yanish, once again, he is a Canadian, lives in Bali, and Phyconomy vocation with commercial seaweed. He has been working on seaweed since 1965, experience all over the world, but he has since then he lives in Indonesia with his Seaweed 6 Energy company. And the next one is Mr. Nelson Padaseri, who worked with Dr. Nish, and he's a co-founder and director of and he's passionate about applying the tools of modern agriculture and biotechnology into seaweed farming, and he leads a multidisciplinary team working on high throughput biomass production from the oceans and developing seaweed cultivation systems in India and Indonesia. Professor Philippe Potang, he is a senior scientist at CNRS in France, and also a team leader of oligosaccharide signaling and defense mechanism at the Station Biology de Roscoff. And he is the scientific coordinator of IDEAL, one of our sponsors for today. Thank you. And of the EU Horizon 2020 SC Blue Growth Innovation Action, GENIAL. He is also a scientific leader of the Safe Seaweed Coalition, a global alliance to establish a seaweed industry that provides safe products, safe working conditions, and environmental protection. The Professor Elizabeth Couture-Cook, she is the head of United Nations University and also Scottish Association for Marine Science Associate Associate Institute. She is currently leading a £6 million research program under Global Seaweed Star. And she is a steering committee of Lloyd Register Foundation, funded a Safe Coalition, a Safe Seaweed Coalition, and several other international working groups. Professor Jana Angadireja, he couldn't be with us today because he's being he suffered from COVID, so hopefully he can wish him speedy recovery. But I think we can keep the questions for him, and interact for those who ask questions for Prof. Jana can be posted in the forum later on. Thank you. I think I ended up there. Pak Mahatma, over to you.
Okay, thank you, Dr. Nana. So before we move to Dr. Kasmiati, let me remind you again that we have time for this session about 1.5 hours. So we have to manage the time wisely. So we allocate for each speaker to respond to the question and to add some more comment about 7 minutes to 8 minutes, no more than 8 minutes. So please keep the time tightly so that we can manage this session, that we can answer all of the questions and we can address all of the issues that come with this in this session. Okay, Dr. Kasmiati, are you there? Dr. Kasmiati?
Yes, I'm here. Yes, Pak Mahatma, I'm here.
Okay, thank you. So, Dr. Kasmiati, many of us interested about what the CEDUS UNHAS has doing, the Center of Excellence of Seaweed in UNHAS. So, another question that we want to know is every country should be working toward a strength repository of all the seaweed it grows, so that genetic diversity is not lost. In such venture, is such a venture being planned by CEDUS and related program?
Okay, thank you, Pak Mahatma and Ibu Nana as facilitator for this session 4. Allow me to greet other speakers. So, good afternoon for speakers come from the same zone as Indonesia, and good morning for others from different zones. So, okay, the question is actually very interesting question. So here, I would like to give some comment on that question. Okay, so the issue of preserve of the genetic diversity of seaweed is important and also interesting, but however, for me, the topic is too big to be conducted by only one organization alone. So here in our organization, we put the program, we plan that program as a long-term goal. So now we are working on the more urgent issues that need to be solved soon as the priority. For example, we work on post-harvest handling and also how to increase the value added of seaweed product. But for this issue of genetic diversity program, we are keen to collaborate with any interesting party, and also beside collaborating between scientists, also government and private sector should participate as supporting agencies to provide financial support for the work. So once again, we are open to have collaboration on the preserving of genetic diversity of seaweed all over the world. So if you have any idea on that issue, please contact us through the email or website. Thank you.
Thank you, Dr. Kasmiati. There is another question that needs to be responded. Please outline lesson to be learned by the other countries with the needs for Center of Excellence. What can you recommend or advise as ways this center becomes sustainable and ensure longevity beyond the initial phase of funding? Go ahead, Dr. Kasmiati.
Yeah, okay, thank you. Here, I would like to explain the Center of Excellence of the Development and Utilization of Seaweed in Hasanuddin University. So, the Center of the Excellence was established in 2011, some years after seaweed cultivation was initiated in South Sulawesi water. And in our experiences, for the first 5-year period, the activities of the Center of Excellence were supported by Indonesian government through the Ministry of Higher Education and also supported by Hasanuddin University. And by then, the center became a well-established organization, and the center has to find its own funding through collaboration at both national and international scales. So coming to this year, we are working on at least 3 collaborative projects funded by Indonesian company and also an NGO from the Netherlands. One of the collaborative project is on the cultivation of porphyra. Porphyra is the red seaweed that with high protein content. The seaweed is promising as a source of food for the future. And another project concern about impact assessment for a large scale of seaweed cultivation implemented in South Sulawesi, including environment, socio-economic, and gender aspects. And also the third project is working with the Indonesian government. The topic is to increase the value added of product based on seaweed. Here, I would like to say that it is an important way to always look for opportunities and also be ready to work with a large range of partners at different aspects of seaweed research and development aspect. Thank you.
Okay, thank you, Dr. Kasmiati. We will back again to you when we have time left. Yeah. Okay. So let's move to another speaker. Professor Nick Paul, are you there? Professor Nick Paul.
Thank you.
Oh, okay. Prof Nick, yeah. I know you are the one who working a lot in Indonesia and you have many partners, yeah. I saw from your talk and your presentation and your video. So I think this question is very challenging question from Alan, Mr. Alan. Yeah, about the creative, innovative product. So the question is, if you had a crystal ball, where will you see the industry being in 10 years' time? And what sort of product being produced and where? Is it clear, Paul? Yeah.
It is, though. Thank you very much, Mahatma. It's nice to be here and yeah, also to represent all of my Indonesian seaweed colleagues, of course, in today's session. So the crystal ball, Dr. Allen, he likes to ask lots of questions. So maybe the crystal ball could be made of a wonderful sort of iota carrageenan, and we could be gazing through that lens. And I think the main thing with innovation in terms of what I've seen from the Indonesian perspective, not the Australian perspective now, just the Indonesian one, is that wonderful entrepreneurial spirit that's happening at the smallholder farmer sort of level. And most importantly, is happening through women's innovation in all sorts of wonderful food and other types of products. So I've never seen such a diverse array of seaweed ingredients being incorporated into every possible savory, sweet, gula type of products that you can imagine. And so that's what I really feel is the great thing about the seaweed production system in Indonesia, is that it is focused on the community benefits. As soon as we start to talk about industrializing that, there's going to be big social and economic challenges that we need to actually deal with. So I'm concerned by that. That's my crystal ball gazing.
Okay, thank you, Nick. Yeah, I forgot to tell that the title of the talk of the Prof. Paul is Innovation and Impact of Seaweed Production and Processing in Indonesia. So one more question before I move to another speaker, Prof. Paul. Can the global market keep taking more and more product?
I know that many of the colleagues on today's Zoom always think about that. What's next? What's going to push the industry into the next sort of level when it comes to actually the pull factors? So we often talk, if you're on the aquaculture side, about the push factors, the production side, but if there's no market, then that's going to become a really big issue. And most of my work is really focused on how do we link into the rest of the global food systems. So it's going to be challenging to get people, and we've done a lot of studies on this from a consumer marketing point of view, to start eating seaweed if it's not culturally relevant. Obviously that's different in Asia, but certainly amongst all other areas. So one of the things that we really need to do is, and Vincent and others have touched on this as well, is start to think about how do we boost agricultural production, existing agricultural production and existing fish and seafood production by linking in seaweed. And really that's the key. We're not talking about replacing protein. We're not talking about, so for feed ingredients, I mean, we're not talking about being the primary nitrogen source for agriculture. What we are talking about is very specific parts of those production cycles. So can we add a very small amount of a seaweed additive into the diet of a fish at a particular stage in the production cycle so that it's most highly valued, so that it can have a big impact? So not trying to be a like-for-like replacement. Can we add agricultural stimulants on a particular crop at a particular stage of a seedling? That's the type of nuance that we need to be looking at, and that's where all of our scientific colleagues should be focusing on those niche high-value products and applications, because that's going to drive the innovation. But we need the research.
Thank you, Professor Paul. Actually, I have one more question in my hand, but let me move to another speaker because of the limited of time. Okay. Okay, thank you, Professor Paul. I will come back to you when we have time. Okay, I move to Mr. Faisan, GV Faisan. Are you there?
Hello, good afternoon. Yes, I'm here.
Okay, okay. Thank you, Faisan. Mr. Faisan, yeah. You are working with Dr. Anisha, yeah?
Yes.
Yeah. And you working about disease and pests in aquatic seaweed. Is that right, yeah?
Yes, sir.
Okay, yeah. Okay, there is two questions for you to respond. One of them is, have you ever identified what are those black spot, not white spot, but black spot on the ecomatoid thallus? Have you seen similar observation in Capapicus? So please, Mr. Faisan.
Yes. Good afternoon, and thank you for that question. Unfortunately, at the moment, we haven't yet identified what those black spots on the thallus of seaweeds are, but we have samples for further analysis. It would be interesting to know the etiology of the black spots and determine whether this abnormality could be a potential problem in the future. Interestingly, we observed those black spots in only one seaweed farms and absent in other seaweed farming areas that we visited. Other than Ecuma denticulata,
We're frozen, maybe.
He's frozen, yeah. It seems he has internet connection problem, yeah. Okay, Mr. Faisan, are you there again? Can you hear my voice? So maybe we skip Mr. Faisan for a while, and then we will back to him again. So we will move to another speaker, Dr. Cecilia Cambe from Malaysia.
Sorry, I lost my connection. Yes,
Oh,
sorry
okay,
about that.
yeah. Okay, yeah. Please go ahead. Continue your
Oh,
comment. Yeah.
I don't know where I left, but other than Eucheuma denticulatum, we have not observed these black spots on other eukematoid seaweed species, including Kapha-Phycos alversi and Kapha-Phycos striatus.
Okay, thank you, Mr. Paisan, about you have clarified about the black spot, yeah. So we haven't found it yet in Kapha-Phycos yet, yeah. But it is potential problem in the future, yeah.
Yes, it's really nice to have these further studies on the potential effect of this abnormality to the seaweed industry.
Okay, yeah, that's good. That's important information to us, yeah. Okay, another question, Mr. Paisan. What mitigation strategy would you recommend to farmers affected by this disease? What the R&D, research and development intervention need to be brought into the predictable seaweed farming?
Okay, yes, mitigating strategies to minimize the impact of disease in seaweed farms is important. Here in the Philippines and many other countries, fixed off-bottom method of culturing seaweeds remain the most widely used farming technique. However, fluctuations in environmental parameters are quite high in the intertidal area or zone, which makes seaweeds prone to stress, thereby making the seaweeds more susceptible to disease occurrence. One of the suggestions mentioned yesterday's session was to move the farm to deeper water areas where fluctuation in environmental parameters are moderate. Also, there are already published papers on the use of seaweed extract, previously mentioned also in the earlier session, as a phycobiostimulant to mitigate disease and pest infestation. The success of seaweed cultivations remain dependent on the prevailing weather conditions, and any changes in environmental parameters could directly or indirectly affect the farm seaweeds. So it's quite difficult to have a predictable seaweed farming at the moment, I would say. R&D interventions and strategies such as identification and or development of new seaweed cultivars and strains that are disease or pest resistant. Also, identification of new cultivars that can tolerate changes in environmental parameters brought about by changing weather patterns and climate change will definitely benefit the seaweed industry.
Okay, thank you, Mr. Paisan, about the information. So it is interesting to know, yeah, this is the lesson learned from Philippines, yeah. Okay, we will back to again, yeah. Okay, now from Philippines we move, yeah, you're welcome. So from Philippines we move to Malaysia. Dr. Cecilia Cambe, are you there?
Yes, I am.
Okay, thank you, yeah. So you did very interesting study about biosecurity aspect of crop care and farm management of the aquatic seaweed farming, yeah. Okay, there is two questions that need to be responded and need to be comment, yeah, further, yeah. One of them is your results indicated that diligent cleaning, cooling, selection, etc., can result in reduced incident of ice syndrome and cleaner crops, hence biosecurity. But what is the return for unit effort? Yeah, the return per unit effort. How does this translate to increased farm profitability, which is probably the goal of most farm enterprises?
Okay, I got the question. So the first for the return per unit effort, we conduct our experiment in the disease season. We call the disease season because that the low water movement around and also the environment parameter in here is not suitable for farming seaweed. That the increased temperature and also the high nutrient in an environment, and also that trigger the epiphyte bloom in that area. So that's why our field experiment is a quite expensive experiment. So the return per unit effort that we get based on our study is required about 2.6 in the biosecurity treatment compared with the non-biosecurity treatment with the cleaning treatment that we made. So about the profitability, I think it's depend on the where we conduct the biosecurity. If they apply in the small scale farmer, individual farmer, the farm just less than 2 hectares, I think that for the labor cost can be reduced because the farmer mostly going to their farm every single day, and then they can conduct the cleaning by themselves. So if the biosecurity for the cleaning treatment conduct in the like large scale or farmer or the more than 2 hectare or farm, it will be costly for the labor cost. But I have to underline here that we conduct in a disease season. So if the farm conduct in the good environment system, I think the labor costs can be decreased because we just conduct maybe it's not every 2 days, but maybe it's less than once per week. So it's once per week or twice per week. So it reduce the cost for the labor cost. So, but if we consistently produce more high quality or the healthy seedling, if in the good season or the bad season or disease season, I think the labor cost can be compromised. I think that's it. My answer.
Okay, thank you, Dr. Cecilia. Yeah, there is one more question. How do you propose we educate farmers that biosecurity measure actually help them into the long run?
Okay, we have to underline here that the biosecurity is a long investment approach. It's not just taking in the immediately have the profit or have the result. So in terms of the how we propose to the farmer to do the such of the biosecurity that we said, I think through the training, even the informal training for the farmer, we know that the farmer is quite doesn't want to show. I think the informal training such as like focus discussion group or any other the community-based discussion can help them to get knowledge about how to do the biosecurity in their farm. Also, we recommend for the government officer that who assist a farmer in their farm. They also can get the access for the biosecurity training because they can assist a farmer with the quick response when there has a problem in the farmer. We also plan to propose our biosecurity practice in using social media such as YouTube or the Facebook to get informed to other stakeholder that maybe it can be reached to the farmer that doesn't have any access to get trained, because when we survey in Malaysia, all the farmer has a mobile phone. So I think with the social media, it can educate them. I think that is my answer.
Okay, thank you, Dr. Cecilia. It is interesting to know. It is good also lesson learned that we know that maybe can be adopted to be implemented to another site, yeah, or to another country. Okay, we will back to again Dr. Cecilia. Let's to move to another speaker, and then from Malaysia we come back to Philippines again. Yeah, Mr. G. Grace Suyo, are you there? Mr. G. Grace Suyo. Do you hear my voice? Miss G, sorry, Grace. Hello, Grace. Dr. Nana, do you hear Grace's voice?
Dr. Grace, can you unmute, please? Or can the organizer?
Hi, sorry, can you
Yes.
hear me? Okay,
Yes,
sorry.
okay, thank you.
Yeah, I think I
Okay.
had problems with my. Hi.
Okay, Grace. Yeah. Thank you. So you are part of the Global Seaweed Star project, yeah?
Yes.
So you talk about keeping afloat stakeholders' insight into sustainability challenges and solution in the Philippine seaweed industry. So there are two questions that we need to know from your answer or for your information. The first one is, in your study, what did you feel were the key intervention needed to help boost the seaweed farming sector in the Philippines?
Well, that is really a very interesting question. So the Philippine sewage industry is currently controlled by market forces, and I think it has also been mentioned by Dr. Ian Nish earlier, if I'm correct. Because even though there are existing policies, implementation and compliance are still weak relative to market forces. And when I say market forces, I'm referring to the law of demand and supply. So in order to strengthen the seaweed industry, there is a need for greater government intervention. So these interventions should actually be geared towards addressing the root causes of the problem, which when we analyze it from a value chain perspective, these issues are actually pooled in the production segment, and this include lack of seed stocks or cultivars, disease and pest infestations, and other environmental issues like typhoons. And in addition to these issues, we are also facing social and economic constraints like lack of access to resources and low compliance to quality standards. So the reason why I am highlighting the role of the government is because it has the executive and legislative powers, and this could be exercised to initiate or strengthen existing strategies pertinent to, for example, quality control and improved access to resources for farmers. And this also entails provision of insurance, credit facilities, which I suggest should always be coupled by technical support or capacity building activities. The government could also increase its control over prices through implementation of price control mechanisms. And one thing that I really would like to highlight is increased opportunities for collaboration between farmers, the government, non-government organizations, the academia, and the private sector. So in the context of social networks, which is basically the relationships between the different stakeholder groups, we can see that the farmers remain at the tail end of the industry because they primarily lack access to resources and services, which can actually be offered by organizations outside of the production sector. So if we want the farming and industry to move forward, we have to make sure that the farmers are able to maximize their access to different forms of support. But take note, but not to the point of making them reliant to external actors, but simply to empower them so that eventually they will be able to manage their livelihoods on their own. And we also need to remember that different organizations have different roles in the seaweed industry. So let me take the Tropical Phyconomy Coalition as an example. So this coalition serves as a platform for scientists, for researchers, for practitioners and entrepreneurs to share their insights about the seaweed industry. But the potential role, potential influence of the coalition actually goes beyond the scientific field, because the coalition can help bridge knowledge and governance gaps in the industry by ensuring that any research output or any initiatives are translated into action. First off, by the government, being the executive and legislative body of every country, and secondly, by key stakeholder groups in the supply chain. So I'm referring to the farmers, the traders, and then the processors. So there's really no definite path by which this can happen, but the research and development sector and the academia can actually serve as drivers for change. And this is through the provision of scientific evidence and serving as a link to funding sources. We have heard those presentations earlier. And yeah, that's from my end.
Okay, thank you, Grace, for the answer and the information. So we will back to you again when we have time. Okay, I will move to another speaker. Thank you, Grace.
Thank
again.
you.
Yeah, you're welcome. So I move to Dr. Ian Nays. Are you there, Ian?
I am indeed.
Okay, Ian, yeah. I know very well that you are very familiar with Indonesian seaweed farmer, yeah, because you work quite long time in Indonesia. So as you know that Indonesian seaweed farmers still practice Phyconomy, adaptive Phyconomy instead of precision Phyconomy, yeah. So the question is, this is a challenging question. Actually, I make this question for you, special for you, because there is no question come from the forum. So I make last night this question to you. So the question is, how is the main challenge faced by the farmer when they are going to integrate the precision technology into their adaptive Phyconomy system for extensive tropical seaweed farming like in Indonesia? Is that clear? Yeah.
Well, and I'm speaking for myself and Nelson Vadasari, who's also on this panel. The difficult part is convincing the farmers that precision techniques are good for their business. And that means spending a lot of time, a lot of effort doing extension work and demonstrating by practical application that precision Phyconomy does enhance their ability to make a profit with their farming operation. I think reference was made previously to tissue culture, be it sort of petering out after 2 or 3 growth cycles. You know, that's the kind of thing that makes farmers a little bit skeptical about precision Phyconomy. So we have to be careful how we integrate precision techniques into adaptive systems.
Okay, thank you, Ian. Yeah, there is one more question. This question come from Dr. Nana. You had been living in Bali for a long time. Do you see any missed opportunity in seaweed business?
In Indonesia?
Yeah, in Indonesia.
I see huge opportunities being missed. I can't even express how big the opportunities are. Beginning with everything east of Timor, Maluku, Papua, all of that, almost untouched. Moving into genera other than the Cumatoids and Gracilaria. Okay, there's a little bit in Caulerpa and that, but Alamenia, there are so many opportunities in Indonesia, I can't even begin to say how big they are. And it applies to the whole coral triangle.
Okay, thank you, Ian. Okay, we will back to you again later, yeah. So we move to next speaker, yeah. Nelson Padasiri. Are you there, Nelson?
Yes, I'm here.
Okay, thank you. So you work actually similar work with Ian, yeah, about the integration of precision technology into adapted economy system, yeah, for extensive tropical rich sea farming. Is that right? Yeah.
Yeah, we work together. Yeah.
Okay. Yeah. One question that arises is, it is very interesting that your goal is similar to those of the Loretta Robertson. This is the first session, yeah. And the question is, do you see any collaboration or cross pollination of idea and efforts? And you will reduce the unit cost of the production so that the value of biomass is interesting for energy production. You mentioned it in your talk, but please outline to the audience the impact of this might have on livelihood of farmer and what are you doing to address this? Please, Nelson, Mr. Nelson, yeah, go ahead.
Thanks for the question. And I think I think there's two parts of the question. First is about similar work being worked on in different parts of the world. And really it is because of the whole TPCD initiative that many of us even come to know a lot more about what everyone else is doing. So I really think there's a huge scope of collaboration, especially when it comes to things about the basic biology and agronomy of the plant. I mean, there's so much to be learned there. If you compare how Kappa ficus or Eucuma is versus, say, a rice or a wheat, we're ages behind that. And we're really trying to use the tools of modern technology to catch up. And rather than all of us working in our silos, I really hope we can, through this initiative, even combine our energies together so we can really bring about, a revolution in seaweed agronomy, right from developing better strains. Then there's been talk about developing hybrid strains and a whole bunch. I mean, all the speakers before me have done such really work. It's great to be here. And definitely there is scope for collaboration, not with many of the groups in this conference. And coming to the second point, really what we're all about at C6. Our motto is disruptive solutions for a sustainable planet, where what we are saying, hey, when we are growing seaweeds, we're not just looking at the current hydrocolloid market, what exists. We want seaweeds to be a part of everyone's lives. So in order to increase utilization of seaweeds, increase the production of seaweeds, we need a lot more uses coming in. And we hear about these uses like, there's bioplastics, there's biofuels, animal feed. I mean, it's all in the background, but for it to really come into the picture, for something like bioplastics to be used by everyone and not just, say, a fancy wrapper on a perfume bottle, for that to happen, we need the price of seaweed to come at a level where these products can be afforded by the general public. I mean, you want every single person to be able to replace, say, a plastic bag with something made out of seaweeds. But to do that, the cost of production has to go down. And for the cost of production to go down, it's not just about arbitrarily reducing the cost, the price of seaweeds. We have to bring about innovation so that for every farmer, it is profitable to grow these seaweeds and still sell it at a reasonable price. The only way this is possible is by bringing in technology. And this has happened in agriculture over and over again. But I think the challenge is, and I think Dr. Phai Kim Lim had talked about it, how to bring about such technology so that it is affordable to the farmer as well. And I really think there's a huge scope for that. Because even in agriculture today, if you see, say, how corn or wheat is farmed, it's really different how it's done in, say, Canada, where people have square kilometers of farms, and a place like India, where even Indonesia, where a few acres is quite a lot. So I think technologies adapt for different farmers, and it's really how do we disseminate these technologies, what business models we use, which will allow for everyone to have a piece of this seaweed revolution.
Okay, thank you, Nelson. Ayan, you are welcome to add if you want to add, because you do similar work with Nelson.
Actually, we don't do similar work. We work together every day. We're like joined at we're doing the same work. So, yeah.
Okay,
so,
thank you, Ayan.
What Nelson says reflects both of us.
Okay, thank you, Ayan. Yeah.
Yeah.
Okay, now we move to another speaker, Professor Philip Potin. Yeah, are you there, Professor Philip?
Yeah, I'm here.
Okay, thank you. So this is about talking about opportunity with the Safe Seaweed Coalition. Yeah. Before we raise the question, we would like to thank you for the sponsor of this event. So we cannot make this event possible without your support. So thank you very much for this support, Professor Philip. Yeah. So now I come to the question. What would you like to see as major outcome from this meeting of the minds and the incredible sharing of information?
So thank you for offering me the opportunity to talk during this workshop. And of course, I really thank you for organizing the whole set of sessions, which were very, very interesting and very stimulating, and which already provided me some answer to your questions, because the main purpose, and I said that in the introduction of my video, is that I really want to push the ideas we developed nearly 20 years ago with Dr. Alan Critchley, but also with Alicia Hurtado and many other colleagues who were involved in the discussion at that moment. And on some of the ideas we implemented at that time in Chile, but we were also willing to implement in Southeast Asia, in Philippines and Indonesia. And so at that time, it was not possible to concretize all the ideas, and due to some difficulties which will be too long to describe here. But during the last 2 decades, I never had to get in mind all the ideas we share. Also, it was, of course, very closely related to my research, and especially my fundamental research, which was based on the biotic interaction of seaweeds and how they cope with different pests. And so that's what I really expect for this workshop, is that we will have provided the opportunity for the community, which is linked to the development of the euchromatoids cultivations and uses for the future, to talk together, and then also probably seed the ideas which will provide new projects for the sectors. And we will, of course, we collaborate with projects between all the stakeholders in the value chain of euchromatoids seaweeds, and especially linking farmers with the scientists, but also with the processors, which are in fact probably key actors in the sector. And what I probably was willing to push 20 years ago was to involve much more the processor. At that time, they were mainly big processors which were located in Europe or in the States. But now we have different pictures. We have also a lot of processors which are Southeast Asia, but also in China. But there is a big responsibility for the processors and the industry, which is really trading the different tropical seaweeds, for really supporting the sector and invest much more than they have done in the past in the key issues for the sectors. So the key issues are really to provide farmers with a security of seeding, with solving their daily problems, and that will not be possible without investment in organizations in different countries. So I'm very pleased to see all the developments which were done in the different Southeast Asian countries, but also in Africa, to better organize the sector and to really invest in the potential of developing some safe solution for producing the seedlings, for considering that the genetic improvement is one very important issue for the future, because without enlarging the genetic diversity of the potential of eukaryotes in the future, we will not solve the long-term issues, and we will never end it with the problem. So all these aspects are important. And what also is important is to see the emergence of new type of industries in Southeast Asia, which are really willing to develop the potential of biorefining for the tropical red seaweeds. And I'm sure that also by providing additional value to the biomass which is produced in Southeast Asia, we will also provide the potential for growing, for make the industry grow. But of course, as now the scientific leader with Vincent Dumézel of the Safe Seaweed Coalition, just is willing to stress that to develop the sector, we will need more cooperation, and we have to really cooperate around the safety issues for the consumers. We have to produce, of course, which will be safe for the consumers, and which will in the future provide new food for the food system, but also to do that in a very large environmental safety. And I'm sure that other colleagues will stress that also in the next coming questions. And of course, I'm considering that the most important person in the sectors are the workers of the sector. So we need to develop a safe system for the workers. And I'm sure that this idea will could be shared by all the stakeholders, and all of us will agree that we have to be safer for the development in industry in the future.
Okay, thank you, Professor Philip. Thank you very much for Ideal G and Genial G. Yeah, I hope that I pronounce correctly. Yeah, Ideal G and Genial G, for making this meeting possible. As you know that we have 40 presentation and more than 500 registrants, so it is huge participant. Yeah, so we hope that the outcome of the workshop will be very productive. Yeah, and also the way to establish the partnership later on. Okay, thank you again, Professor Philip. Now I will move to so maybe I think this is the last speaker that we want to ask, Professor Elizabeth Cotter-Colt. Yeah, hello, Professor Elizabeth. Yeah.
Hello there. Hi.
Yeah. Okay, we want to know the interesting thing about the global seaweed star. Yeah, so this is building a progressive management pathway for the seaweed biosecurity. So there are two questions that need to be respond. One of them is, so this is from Alan. What happened after the funding finish? What are the major part of the global seaweed star legacy?
Sorry, could you repeat the first part of the question, please?
Okay, sure, yeah. What happened after the funding finish? What are the after the funding finish? Yeah. What are the major part of the global seaweed star legacy? Yeah.
Okay, thank you so much. I'd just like to start by thanking everyone and thanking the organizers for inviting me to come along and speak to you all here. I'm currently speaking from the beautiful Isle of Man off the west coast of Wales, but thank you to everyone who's invited me. So yes, so Global Seaweed Star is a 4-year program funded by the UK Research and Innovation Fund. And it's due to come to an end this December. Now, the plan is that we look for further funding, and we did actually apply to the European Union to one of their funding calls, which actually allowed partners from Southeast Asia, it allowed international partners to take part. So that was submitted in January, and that was going to be a 17 million euro program. But unfortunately, we found out recently that that wasn't going ahead. It was and it would have included a number of people who are here in this meeting and part of the Safe Seaweed Coalition as well. So that was a real shame. Unfortunately, due to the COVID pandemic, funding has become a lot tighter across the world, as everyone realizes. So there is obviously other funding opportunities that are going to become available, such as the Safe Seaweed Coalition. And obviously I'm on the steering panel for that coalition. And I've been hosting, I've already hosted one stakeholder sort of round table on environmental standards and biosecurity standards. So hopefully there will be further funding for the Global Seaweed Star Partnership or partners within that to go and acquire sort of further funding from the Safe Seaweed Coalition. But there are other funding opportunities, and I'm hoping that next year, I'm not sure whether you're aware, but the UK government actually cut its overseas aid budget this year, but there's been a lot of us lobbying very hard for that to be returned to the level that it was before the cut. And hopefully there will be more types of funding through UK research and innovation that will allow us to work with countries in Southeast Asia again. So I certainly have many ideas. I still have ideas, and hopefully I'll be able to recycle the European proposal, which was looking at upscaling seaweed cultivation to try to look at how it can increase biodiversity, restore degraded environments, but also obviously provide economic benefits for the seaweed farmers themselves. So there are other opportunities to continue on, and I'm sure there will be many of us from the Seaweed Star group. I know a number of us are now in the Safe Seaweed Coalition, so we will still be together. And, you know, in terms of the legacy, Alan, this is a great question, thank you. In terms of the legacy for Global Seaweed Star, well, obviously there is the legacy of, I mean, we've already published 17 scientific publications, and there's going to be probably another 30 at least come out from the program. So that's a legacy in itself. But we've also really raised the profile of biosecurity, particularly within national governments. A lot of people didn't really understand what that meant, but we have raised the profile of that to the extent that, thankfully, you know, and it's a wonderful opportunity. I'm working with Melba Renazzo at the FAO in the aquaculture department to develop this progressive management pathway for the seaweed aquaculture industry. And I'm hoping that a policy document will be written, an international policy brief will be written by the end of this year, which hopefully will then be a physical piece of paper that policymakers can take to their ministers in every country growing seaweed and sort of say, look, these are the key recommendations, and this is what we should be following to make sure that our seaweed industry has the best possible chance of succeeding. So there are hopefully going to be many, many more legacies and many more impacts that we haven't even really thought about yet. But those are the key ones anyway. Hopefully that answered your question, okay?
Okay, thank you, Elizabeth, yeah, for the information. Yeah, there is one more question. Yeah, so this question also comes from Dr. Alan. So he said that you have built an incredible network of scientists, scientist economists, yeah, and undertaken considerable training of the next gen economists. These people are now based in some of the major centers of seaweed production. How can that talent be used to help countries that we are not in the first phase of funding?
Okay, well, thank you very much, Alan, for saying that. That's very kind of you. Yeah, I mean, it was really a fantastic opportunity, actually, because part of the £6 million that we got from UKRI, nearly 1 million was put aside to allow us. It was we created this Global Seaweed Star Research Fund, and we also had a travel fund. Hopefully some of you benefited from that. So it allowed us to basically fund 15 additional projects in seaweed producing developing countries in other parts of the world, other than our core partners, which I should have said were Tanzania, Malaysia, and the Philippines. So we've worked with many other researchers in different countries around the world as well, and also helped over, I think, 100 researchers now through the Travel Fund get to various different conferences. So yes, we have built up this incredible network, which is wonderful to see. And I'm hoping that, well, and we're very much encouraging all of those researchers to join with the Safe Seaweed Coalition, because I really see that as the next sort of grouping, lobbying group, as it were, that we can all work together and collaborate with. So I'm hoping that many, and really urging many of our partners to join with that coalition. And of course, keep in touch through the Global Seaweed Star website and email address, so we can try to identify funding opportunities where we can work together. And I think, I'm really looking at other new forms of funding. So a couple of years ago, I applied for the MacArthur Foundation. So other sort of maybe US-based philanthropic funding organizations that could maybe be used to provide to fund another Global Seaweed Star type coalition. Yeah, so hopefully that answers your question. Thank you.
Okay, thank you, Professor Elizabeth, for providing us information more about Global Seaweed Star. Yeah, thank you. So now, yeah, you're welcome. Now I move to Dr. Nana. So maybe we have a question in the Q&A box. So please raise the question. So time is yours, Dr. Nana, please.
Yes, thank you, Dr. Mahatma. Actually, we have a question for Professor Jena Angadireja. Unfortunately, he's sick from COVID, so wish him best speedy recovery. The question is about, the question is, at current cost of production, is it reasonable or feasible or economic to grow seaweeds to use for biofuel and or sink the abyss for carbon capture? And we invite anyone among the speakers who want to answer that question. Maybe we can begin. Who wants to volunteer?
I will.
Thank you. Yeah,
it's Ian. Well, I've known Yana since what, 1988. So you'll have to excuse me for answering. I think, Nelson addressed the issue. Before it is remotely feasible to use seaweed as a raw material for biofuel production, we need to find a way to get the cost of seaweed production in massive quantities down to a level that is about, let's say, 1/3 or less of what it is now. And at that level, it still has to be profitable for the farmers. This is, Nick, I see you there. What do you think? I mean, do you agree with that?
You want to get me started about biofuels, Ian? So,
well, yeah, go ahead.
After you've taken the protein, taken the fatty acids, taken every last possible thing, if there's a tiny little bit of carbon left over and you think you can make some biogas from that, go ahead. But it's a long way before that will happen. So,
yeah,
and of course, carbon capture is the next level down from that. So
yeah, and the other thing that came up earlier was that if it's just a growing seaweed and then sinking it to 2 kilometers down to sequester carbon. And the question is, while you're sequestering the carbon, what else are you sequestering? You grow seaweed that is very valuable. It's absorbing CO2, but it's also mining many other elements from the seawater. And then you sink the whole thing to the bottom of the sea. How does that even remotely make sense? I mean, it makes biofuels look like a great idea. Simon?
Anyone wants to
answer to
comment
that?
on that?
Yes.
Nelson?
I would say there's another way for carbon sequestration, and it's a bit indirect, but through biostimulants alone. What we found, I mean, we also make biostimulants, and what we found that, hey, if you grow seaweeds and you measure the amount of carbon captured in seaweed alone, versus if I take the same seaweed and I make it a biostimulant, and then I spray it on virtually any crop, because the amount of the amount of seaweed which maps onto an area is so huge. Even if you get a 5% increase in productivity, that's a massive amount of carbon which gets captured. And why I say that is most farmers only look at productivity in terms of the crop you get. Like if I'm growing corn, then I measure the amount of corn I get. Nobody bothers measuring the increase in the plant mass, the increase in the root mass, which is all really the carbon capture. So I think until really we can get the cost of biomass really, really low, biostimulants and putting the biostimulants on crops to me is a great way to sequester carbon in a completely organic manner with no chemicals.
Yeah. I mean, people keep talking about growing these massive amounts of seaweed over the ocean. What you have to understand is that all seawater is not created equal. The distribution of nutrients is very, very, very unequal. If people look at seawater and all water looks the same, but seaweeds have been, macrophytes have been around for 1.2 billion years, fine-tuning their capacity to uptake all kinds of elements from the seawater and translate it into growth. To me, that is the big mystery that we have to solve, is how to deal with the nutrient distribution situation.
Thank you, Dr.
And of course, biosecurity enters into that whole thing. I think I'm still trying to understand what biosecurity means and how we explain it to seaweed farmers in Southeast Asia, but I'm working on it. And your paper was thank you for that. It explains a lot, but I'll be getting back to you with some questions if you don't mind.
Well,
okay.
And I'd like to add one thing here.
Yes,
Nelson. Talking about large farms. Today, all of seaweed farming is really seaweed farming on the surface. So we're extracting the nutrients which are coming in. There's no input really. But as we grow in scale, and there's a lot of R&D needed for this, these are just in concepts. There are ways in which these nutrients could come in to actually boost seaweed growth. One, I think Brian has mentioned, you could use natural upwelling, where there's a lot of nutrients below the sea surface, which could which could actually increase the growth. And what Nick pointed out, extracting all forms of carbon out and what's left going into byproducts. What I say is, if you focus only on the carbon, what's happening to all the nitrogen and all the salts which are present in the seaweed? So as you grow in scale and you start utilizing the carbon more and more, what we may find out is we could actually use those byproducts, these elements present in those byproducts, to fertilize the fields. Now, here is, these are really great concepts to talk about. There's a lot of work to go in before these actually happen. And yeah, maybe I'm sure a lot of collaborations would probably lead the way there.
Thank you, Nelson. Professor Philip and Professor Elizabeth, do you want to say something? Comment on this one?
Yeah, I was willing to comment that, of course, we have to be, as we were discussing previously about the difference between micropropagation and tissue culture. We have to be very cautious with the wording about carbon sequestration. So seaweeds are indeed storing carbon. So and they are very efficient to store carbon when they grow, but they are also releasing a lot of carbon during their growth. There's a life, and so that means we have to consider that also that this carbon will be recycled in the systems, and we need to really be cautious about that. And then in terms of sequestration, we will start to sequester carbon if we are intelligently using seaweeds. So of course, using seaweeds as we do with other biomass in material which could be kept on a long term, and which will provide potential for new materials. We will save also oil-based products, but also the potential to develop the sectors which are at the moment very impacting in terms of greenhouse gas emissions, such as in agriculture. So of course, it was already discussed about fertilization, and also the potential to mitigate to get protection of crops using seaweed products. So that means in that term we will be efficient in a long term to solve a part of the carbon issue at the moment. So, but seaweed will not will not be the sole solution to solve the carbon sequestration problem on Earth. And we have just to contribute and to provide demonstrations that it is part of the solution. So that's very important. And if it helps to support the development of the sector, we have to, of course, to encourage that. And we will consider all the projects will be reasonable in terms of improving this in the future. And that's what I was willing to stress at the moment, because we need to stay science-based. In fact, because we are simplifying too much the system. So as it was already demonstrated by Ian, so in terms of nutrients, of course, seaweed could not be grown everywhere. Even if we have the best engineering for restoring the upwelling, I'm not sure we will be able to put seaweed everywhere. But we will be able to develop the cultivation, and that this cultivation will provide new services for remediating some of the decline in many oceans. So that's the solution for the future. So thank you very much.
Thank you, Professor Potang. Unfortunately, we already reached the end of this session, and thank you, Dr. Mahatma, for facilitating this session. Let me close this session. So, how do we move forward? Global collaboration and partnerships are a necessity to find solutions to the global complex global challenges we all face. And this is a kind of consensus among all the speakers upon hearing this 2 days workshop, as well as the online presentations. And it has to be done in such a way that is ensuring sustainability. And among the many issues and challenges in seaweed business, where do we begin? How can we tap on limited resources we have for the greater benefit to mankind and the health of our planet? Surely this is just the beginning of this kind of partnership, and we hope to see many, many more exciting forums like this and many more collaborations to come in the future, capitalizing the different strengths we all have and we have seen throughout this 2 days workshop for stronger coalition in seaweed business. So it is the emphasis on collaboration and cooperation rather than competition. And we certainly encourage all our audience here to join the Safe Seaweed Coalition as well, to be part of exciting revolutionary journey. The complex global challenges we face needs concerted global efforts, and it is framed in SDGs as our common goals, as emphasized particularly by Dr. Vincent Demezo. So how can we feed the increasing number of world population from the sea? How can we feed the increasing number how can we find materials that are environmentally friendly? How do we balance between the needs for economy and the health of the ecosystem? These are among all the key questions that whose answers require global effort. In doing so, we leave no one behind, because after all, we live in a connected world, in a connected sea. So let's connect our science, let's connect our research, let's widen our partnership beyond our national borders, stronger together and more resilient in facing uncertainties the future may offer. So ladies and gentlemen, friends and colleagues, with this session 4, the very last question of the Tropical Phyconomy Coalition development is concluded. Thank you very much for all your participations, commitment you made working at odd hours due to the time zone difference. So thank you to all our sponsors as well, GENIAF and IDIAF and the Universitas Hasanuddin for hosting this event, and hope to see you again soon. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon and good day. Thank you.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you.
Thank you.
Thank you, everyone. Thank you for
Thank
the
you.
excellent speaker. Thank you for the distinguished participant. Thank you, Dr. Nana.
Thank you, Pak Mahatma.
Thank you, everyone.
Right. Hello, ladies and gentlemen. It has been a great honor for us to host so many seaweed experts and such a wide seaweed constituency from around the world. I would also like to thank all the team here and abroad who have contributed so much to making this event happen. On behalf of the Universitas Hasanuddin Executive Committee, I would like to thank you all for your valued contributions to this lively workshop. Please remember that the forum is live for continuing interactions and that there is still time to get your certificate by posing a question on the forum by 15th of July. I now pass the screen to Professor Jamaluddin Jompa, the UNHAS TPCD-1 Chair and Dean of the Graduate School, to make some final remarks. Over to you, Prof. Jamal.
Hello, everybody. Can you hear me well?
I can hear you. I hope everyone else can.
Okay.
Yes, we can hear you.
Thanks again for everybody. Finally, this is the time to bring closer to our very intensive 2 days of meetings. I have problem with, but I hope this is also not the end, but the beginning of our opening of a new era of research. Hold a second. We have
Is this better? Hello?
I could hear.
Is this better?
Can you hear me? Oh no, I can't. Sorry, I cannot hear you.
Hello?
Maybe you better come and sit at this laptop and talk if you have a problem with that one.
Okay. It was really clear to begin with.
Yes.
Maybe Ambi, can you put
perfect.
off your speakers? Hello?
Yeah,
We
he's
can hear
coming
you, Prof.
through
JJ.
fine. I can hear him.
Okay, so let me just repeat again that I do hope this is the beginning of our opening of a new era of research and collaboration among us. Hello?
Yes, we can hear you fine.
Gotcha.
Yes, all of us on the ecomatoids coming together to address all of the issues that we have heard are so well presented in this 2 days of workshop. Of course, our thanks go to the presenters who gave us so willingly of their time and talents. Without you guys, this workshop would not have been possible. Your works remain online. I watched many of them, almost all of them, and we encourage continued chat in the Tropical Phyconomy Coalition Development Forum. Thank you also to all the enthusiastic audience. You guys great. The future lies in your hands. Please grasp these opportunities. Please feel free to reach out the panelists and the Center of Excellence, which are dedicated to ongoing research and tropical seaweed and economic solution to improve coastal environment and location. Sorry for my voice. Thanks goes to also UNHAS moderator, facilitators in the last 2 days. Let me just call by their nicknames: Pak Rijal, Mbak Dila, Bu Arti, Bu Elmi, Pak Sudi, Bu Inaya, Bu Nana, and Pak Mahatma for your skillful fielding question and keeping us on time. I know it was challenging, especially in this, sometimes we have hiccup technology, which is also understandable. You guys did excellent job. Thanks again. I also would like to thank all the people behind the scene, operators. These webinars do not happen by themselves. and we know this technology soon need a lot of skill adjustment. In particular, many people in the front, but also behind the scene, including Abigail, Widya, Amanda, Ihsan, Iqbal, Mukhlis, Tamen, and so on. Also the volunteers of Nelson Farisari and also R.A. Narayanan from the C6 Energy. We also thank Director of UNHAS and Graduate School of UNHAS for the facilities, and also MARSEF for being the umbrella of organization for this meeting. MARSEF is under the Faculty of Marine Science and Fisheries of UNHAS. Our special and sincere thanks, especially to our sponsors of this workshop, which are RDLG and GNLG, as administered by Dr. Philip Potin, to the station of Balikur Roscoff, France. We could not have done this without your support. Please continue to collaborate with us for the much more needed research and effort, as well as please continue to support this excellent platform. This is a very inspiring name, by the way, TPCD, Tropical Phyconomy Coalition Development. I like the name. Our special thanks also again to TPCD-1 initiators, Dr. Ianis, Dr. Alan Critley, and also Dr. Nelson Palaseri and Abigail. I know many of this meeting I attended. We need to be adapting the time zone to discuss all of the preparation the last a few months. I also have been asked to remind all presenters they will be being contacted for a possible submission of chapter for volume 2 of the tropical seaweed farming Phyconomy by the team of Hurtado, Neis, and Critley, and I think in the very soon you will be contacted. Finally, we come to the end of the TPCD-1. It just remains for me to state that the euchomatoids are only part of the growing family of cultivated tropical seaweed. Here in the Coral Triangle, as you know, we are all placed to produce seaweeds for the world. And there are many more seaweed groups, as we all know, Gracilaria, Sargassum, Colerva, Ulva, and so on, requiring the same type of attention as the euchomatoids in order to fulfill their greatest potential to provide goods and services. There will be forthcoming workshop dealing with this seaweed. So guys, please watch this space and be willing to participate in the next workshop. I do hope the next workshop will be more participant, more enthusiastic, and I think we need to be moving on from this pandemic. Indonesia now has faced again the third wave, so but we need to be enthusiastic in finding solution for the world. I hope you all stay safe and healthy, and we'll see you again in the not too long distance which in this channel. Again, thank you for and all, and I formally declare this workshop closed. Terima kasih. Wabillahi taufiq wal hidayah. Assalamualaikum warahmatullahi wabarakatuh. And take care.
Thank you.
Thank you very much, Professor Jamaluddin Jampa, for those inspiring words. We now wish to leave you with a taste of South Sulawesi, even though you can't come here. Once again, we will share with you this time a traditional Buginese song, which is called Marencang. It is performed by the Hasanuddin University Social and Political Science Student Choir, the DB3 Voice. This performance won the gold medal in the folklore category at the 3rd World Choir Festival in 2021. Stay safe, healthy, au revoir, and sampai jumpa.
Sama-sama.
Thank you.